Harper Audio presents The Science of Jurassic Park and the Lost World, or How to Build a Dinosaur, written by Rob DeSalle and David Lindley, read by David Lindley. Take a seat. The tape's in the VCR, the popcorn's at your side. You're watching Steven Spielberg's movie of Michael Crichton's best-selling novel, Jurassic Park. Across the bottom of the screen, a caption tells you where you've been transported to. Mano de Dios Amber Mine, Dominican Republic. As you get drawn into the story, you imagine yourself part of the action, hanging around with the crew as the movie is being made. You're on a rickety raft being pulled across a river to a muddy landing area. You cautiously step off the raft and find yourself in the middle of what appears to be a large, shallow excavation dug out from the surrounding dense forest. In the small cliffs that ring this quarry, you see timber-framed openings. Through them, miners are coming and going with baskets. You move forward to investigate. Now you're in one of the caves in the cliffside. Around you, local workers hack away at the cave walls, looking for pieces of amber. Mr. Spielberg? Sorry to interrupt, but you're not likely to find any amber here. Dominican amber mines are mostly up in the hills, away from the rivers. No jungle. More like a sort of open scrub, you know, little trees and shrubs. And most of the amber mining isn't done in caves. No, I guess you're right, it wouldn't be nearly as scenic. But we want to do things right, don't we? We just have to move everything a few miles, okay? Now you're away from the river, up on a hillside instead. Miners hack away at the ground with shovels and machetes. Among rock fragments, the practiced eyes of the miners pick out darker flecks. These are pieces of petrified wood looking almost like coal, and in them the amber is often found. As the miners dig and pick over what they find, one comes toward you with an unusually large piece of amber. Cleaning the surface, the miner holds his find up to the light. There, embedded in bright yellow amber that seems to glow still with sunlight from the time it was formed, is a huge, perfectly preserved insect. Oh, Mr. Spielberg, I'm really sorry to interrupt again. Dominican amber certainly has insects in it, but they're not going to be any good for what we want. They're about 25 million years old, maybe as much as 35 million years old. That's just not old enough. Dinosaurs died out 65 million years ago. So this little insect may have some sort of animal blood inside it from the last time it fed, but it can't be dinosaur blood, you see? No, I'm afraid all the amber in Central America is pretty much the same age. I remember reading just last year the scientists at the American Museum of Natural History in New York made this wonderful discovery, a mosquito trapped in amber that's at least 85 million years old. Where? That's the bad news, Mr. Spielberg. They found it in New Jersey. But we really do want to get this right, don't we? New Jersey it is, then. It's a sweltering East Coast summer. The noise and fumes of traffic and construction machinery are all around you. You're in a field up to your ankles in thick, gluey clay. You dig up a spadeful of the heavy clay and rummage through it by hand. In an afternoon of back-breaking work, you come up with a couple of dozen small pieces of amber. Examining the pieces through a microscope, you see countless little midges and tiny worms, but bigger insects are rare. Sifting through the 80 pounds of amber that you've collected over the summer, you've only come across one mosquito. Still, you finally found what you were looking for, a biting insect 85 million years old. And it may, you hope, even have sipped the blood of dinosaurs. Now, you just have to open this nugget of amber and see what you've got. Say the words Jurassic Park, and people instantly know what you're talking about. In the story, scientists bring dinosaurs back to life. The dinosaurs turn out to be smarter and scarier than anyone suspected, and mayhem ensues. You could call it just another replaying of the old Frankenstein saga. Well-intentioned scientist creates living creature, creature becomes monster, monster goes on the rampage. But the extraordinary success of Jurassic Park testifies to Michael Crichton's ingenuity in adding a couple of fascinating new twists to that story. First, there's the idea that bringing dinosaurs back to life may one day become science fact. In 1912, Arthur Conan Doyle wrote a fantastic story called The Lost World. In this tale, an intrepid scientist traveled to darkest South America and came across a lost plateau high in the Andes where dinosaurs still lived. In using the same title, The Lost World, for the sequel to Jurassic Park, Michael Crichton recognized that nowadays beasts and wonders are more likely to come from the unknown realms of science, not from any lost place on Earth. Modern scientists have already recovered bits of ancient human DNA from Egyptian mummies and from an unfortunate fellow who died in the Alps thousands of years ago and got trapped in ice. A number of scientists have succeeded in extracting DNA from insects and plants that died around 30 million years ago. If these remarkable discoveries are possible, is it so unreasonable to imagine that some years from now, dinosaur DNA might not be coaxed out of some ancient remnant? And second, of course, there are the dinosaurs themselves. Tens of millions of years ago, dinosaurs ruled the planet. The biggest dinosaurs were terrifyingly large, and some were undoubtedly ferocious predators. Just to think of those creatures treading the same ground we walk upon today inspires awe and, perhaps, a sense of our own fragility. From the first discovery of dinosaur fossils in the early 1800s, there was an irresistible tendency to think of them as dull, lumbering giants, dominating the earth by brute force. In the Victorian era, people could feel comfortably superior to these extinct brutes. Earlier forms of life were by definition failures, rough drafts of things to come, and humanity was God's finest handiwork. But in the last few decades, all that has changed. Careful excavation of fossil sites around the world have yielded the remains of dinosaur nests and youngsters, forming family and social groups. Although it was often assumed that dinosaurs were a dead end of evolution, many scientists believe that the birds, which must surely be counted among the most successful creatures on Earth, are the distant descendants of one particular branch of the dinosaur family. It used to be thought too that dinosaurs were cold-blooded, like lizards and snakes and crocodiles, and with all the psychological implications that cold-bloodedness carries. But now there's a substantial body of opinion among scientists that dinosaurs may have been partly or completely warm-blooded. Finally, any comfort we might have taken from thinking that the dinosaurs died because of their stupidity or clumsiness has been upended. It's almost universally accepted now that the dinosaurs were killed off by a catastrophe they could do nothing about. A huge meteorite slammed into the Earth, sending so much dust and debris into the atmosphere that the warmth of the sun could have been largely blotted out for many years. In the ensuing dark, the dinosaurs all died. You could hardly say that the dinosaurs were unsuccessful. Their 160 million years of existence testifies that they had established a comfortable and prosperous existence on the prehistoric Earth long before we came on the scene. We may count ourselves lucky if we can stick around for even a fraction of the time that the dinosaurs survived. But will scientists really be able to recreate dinosaurs one day? If you can find the DNA of a dinosaur, have you not found its genetic blueprint? The idea of making a dinosaur may be altogether speculative, to be sure, but it seems like a speculation built on genuine scientific achievements and knowledge. On the other hand, you don't have to be a scientist to guess that there might be a bit more to it than that. A lot of fundamental scientific problems would have to be solved before the idea of turning dinosaur DNA into an actual dinosaur could even be usefully tackled, let alone accomplished. What's more, the task of building a dinosaur is not one big problem, but a host of problems, small and large. Some little detail, some tiny piece of missing information could undo the whole plan. Even if the science of Jurassic Park, at least in broad outline, is based on fact and on plausible guesswork, that doesn't guarantee you will be able to solve all the problems that might come up. But then again, although any number of these problems might be impossible now, it's not clear that any of them are impossible in principle. We don't claim that when you get to the end of this book, you'll know how to make a dinosaur. But you will, we hope, have a better idea of what sort of problems stand in your way and how much work you'd have to do to solve them. So you want to make a dinosaur? First things first then. As far as anyone knows, the stuff dinosaurs were made from was no different from what all other living creatures are made from. Proteins, fats, carbohydrates and sugars, lots of water, a dash of salt, various other minerals here and there. What you really need is the set of instructions for making a dinosaur. A blueprint, if you like. To scientists, that blueprint is known as the animal's genetic code, its DNA. You have human DNA in just about every cell of your body. You could get a sample of your own DNA from a mere scraping of skin. But how do you get DNA from an animal that became extinct 65 million years ago? In Michael Crichton's story, the character Louis Dodgson, he's the dishonest product development chief for the Biosyn Corporation, hears rumors that the rival scientists at the InGen Corporation have gotten hold of some dinosaur DNA, and he speculates that somehow they must have extracted this DNA from fossils. It used to be thought, he muses, that fossilization eliminated all DNA. Now that was recognized as untrue. Is Dodgson onto something? Could the InGen scientists have got DNA from fossils? The museums of the world contain all sorts of broken remnants of fossils, useful to researchers but not so interesting to the average museum goer. Why not just gather together a few truckloads of dinosaur fossil fragments, grind them all up into powder, and see if some DNA didn't get preserved somewhere in them? Is it too much to ask that in a ton of fossil fragments, just one copy of the DNA could have survived unchanged? As a matter of fact, the answer is yes. It probably is too much to ask. Most of the time, when an animal dies, its remains don't stick around for more than a few months. Nature is an efficient recycler. Dead animals get turned back into soil, plants, and other animals, which die and then contribute to the next generation. Occasionally, under certain conditions, decomposition can be staved off for a while. In Yellowstone National Park, it's not uncommon for the spring thaw to uncover from the melting snows the remains of bears that died just as winter was setting in. But once the snow melts, nature begins to run its course. Is there a way a dead dinosaur could have stuck around longer without decaying? In September 1991, 2 hikers in the Austrian Alps came across what they thought was the partially preserved body of another hiker. But this was an oddly dressed hiker, wrapped in rough furs and wearing what appeared to be laced sandals of animal hide. Forensic investigation revealed that he had lost his way about 5,000 years ago. Ötzi, as the unfortunate alpine iceman was named, may have died in an unlucky accident, but the almost perfect preservation of his frozen body was a piece of luck for modern scientists, who could figure out not only what kind of animal skin he was wearing, but what he had eaten just before he died. More than that, scientists were able to extract a few bits of DNA, which turned out to be pretty much identical to DNA that you would extract from any modern inhabitant of Northern Europe. Even older than Ötzi is a deep-frozen woolly mammoth that was found in Siberian ice some years earlier. Mammoths became extinct in the Pleistocene. This creature is reckoned to be about 40,000 years old, and its skin, flesh, and fur were remarkably well preserved. Here and there around the world, conditions have allowed human or animal remains to last for an unusually long time. In a peat marsh in Cheshire, England, a 2,000-year-old human body, leathery but recognizable, was found in 1983. This character, who became known as Peat Marsh, was preserved because the heavy murk of the peat bog had kept out oxygen, which helps prevent decay, and the acidity of the bog tended to kill bacteria and effect chemical changes in the flesh which made it less prone to decomposition. Peat marsh was pickled, in other words. Similarly primitive Europeans, hundreds of them in fact, have been found preserved in bogs throughout Denmark and Germany as well as the British Isles. The oldest example of preserved human DNA comes from 8,000-year-old human remains found in Windover Pond, an acidic bog in Florida that was apparently used as a burial place by early Floridians. All very intriguing, but even if a dinosaur had gotten pickled in a peat bog 100 million years ago, the peat bog itself wouldn't have lasted until now. For the remains of a dinosaur to have lasted to the present day, another sort of process has to occur. That something is mineralization, and it's this process that creates fossils. Bones can survive for a long time in the heavy, airless mud at the bottom of a swamp. And sometimes, when conditions are right, the squishy sediment containing the bones of our dinosaurs gets increasingly compacted by the weight of further sediment pressing down upon it. Over the course of millions of years, sediments get compressed into sedimentary rocks, chalk, limestone, sandstone, and the like. The same slow changes that turn sediments into rocks can turn the bones within the sediments into petrified rock-like remains. Fossils, in other words. As water trickles through the sludge and the bones within it, anything that can be dissolved, no matter how slowly, gets replaced by insoluble minerals. Because bones are porous, water can seep through them, leaching out the organic content and filling the spaces with mineral material. Because the process is so unimaginably slow, taking thousands of years, the replacement of organic by mineral material proceeds almost one atom at a time. The structure of the original organic material is preserved, but its chemical nature is completely changed. What used to be bone has become, quite literally, stone. But that also explains why you're not likely to find any DNA in a fossil. The DNA is part of the organic content that got replaced by rock. If we need a dependable way to find dinosaur DNA, we had better abandon Louis Dodgson's idea and look again at what the scientists of Jurassic Park did. Mosquitoes and other biting insects have been around on Earth for a long, long time. Imagine a mosquito biting a dinosaur and drinking a little of its blood. Hold on a minute, could a mosquito really pierce a dinosaur's skin? Occasionally, fossilization has preserved not only bones, but also fragments of tough skin or the imprint of dinosaur hide on thick mud. It's not too far off the mark to think of dinosaurs as having something like alligator hide, which is tough enough to keep mosquitoes out. But even so, there must have been softer, more flexible skin around its eyes and mouth. We can never be sure that insects actually fed on dinosaurs, but they may have. Let's pick up the scene again. After taking a sip of dinosaur blood, our mosquito flies off to rest on a tree and digest its meal. But it happens to land on a tree that's oozing gum. The insect's feet get trapped in the sticky gum, and as the gum flows, it rolls over the luckless mosquito, covering it completely. Exposed to air and sunlight, the gum begins to harden, and after a matter of hours or days, it has solidified into a resinous mass. This is amber. It's largely an accident of nature that amber happens to be such a remarkable preservative. Tree resin is basically medicinal. It hardens over damaged bark to protect the tree as it heals, and it contains antibacterial chemicals that keep decay and disease at bay. The antibacterial qualities of the resin help to preserve the corpses of trapped insects. It also turns out that amber has powerful desiccant properties, that is, it soaks up water molecules and locks them away within its chemical structure. That quality happens to be particularly useful for preserving DNA, which tends to break up in the presence of liquid water. All these properties of amber explain why it's an excellent preserver of insects. But only a few kinds of tree resin harden into amber that will last thousands or even millions of years. And even these ambers have to be protected. In air, they absorb oxygen, which causes them to gradually weaken and crumble. Even the most durable ambers, if they're to last 1,000,000 years or more, have to be buried somehow. One likely scenario is that when trees carrying their amber jewels would die and fall to the ground, they embed their impervious fragments of amber in heavy muck. In the Dominican Republic, pieces of amber are trapped within wood that has turned to lignite, a kind of coarse coal. But as we discovered, Dominican amber is not old enough for our purposes. We need to go somewhere else. New Jersey will do fine, although parts of northern Canada will fill the bill too. If you want to insist on an exotic location, we can take the film crew to Lebanon, where there's amber that's 125 million years old. At any rate, we don't really care where the amber came from, as long as it is at least 65 million years old. Sifting through this amber is no easy feat. Finding it in the first place takes a certain amount of expertise. A piece of amber in the ground won't look very remarkable. It will be dirty, coarse, scratched and dull. The early scene in Jurassic Park, in which a miner holds a nugget of amber up to the light to show off the big bug it contains, is a touch of cinema faux. For many pieces, it's only when you get the amber back to the museum, where you can clean it up and polish it, that you can start looking for insects. Amber mining and collection is quite an industry in several places around the world, so John Hammond, the fabulously wealthy entrepreneur of Jurassic Park, doesn't have to get his hands dirty by digging holes in the ground himself. He first comes to the attention of US government officials because he's spent $17 million stockpiling amber from sources all over the world. He even buys up amber jewelry, amber ornaments with bugs inside are a popular novelty item. This is something that scientific amber collectors would never do, because you would have no way of knowing how old a piece of amber is. To know its age, you have to know what kind of rocks or soil it came from. Dealers only want to know how good the amber looks and how much they can sell it for. John Hammond's problem, however, is that he needs all the amber he can get to search for the handful of pieces that contain what he's looking for. Even if you find a piece of amber with an insect in it, the chances that it has dinosaur blood inside it are small, so Hammond wants, in effect, to corner the market. So we're left with New Jersey, Lebanon, Canada, and a few scattered sites in Europe and Asia. Let's just assume that in among all the amber that Hammond manages to buy, there's a good deal that could be old enough to contain insects that might have bitten dinosaurs. Now it's just a matter of sorting through it all. That's all very well, but what have scientists actually found in amber? There are plenty of plant fragments. More spectacularly, there have been discoveries of spiders, scorpions, even whole frogs. Little frogs, of course, no more than an inch long. But what about insects? New Jersey amber is rich in insects, but the great majority are insignificant gnats and midges. There's the 85-million-year-old mosquito, recently discovered in New Jersey amber by scientists at the American Museum of Natural History. And a nasty bloodsucker called a sandfly was discovered in a piece of Lebanese amber, 120 million years old. On the other hand, no one has yet found a preserved insect with its stomach full of blood. Nevertheless, the first part of the Jurassic Park problem, finding a suitably old insect with dinosaur blood inside it, doesn't seem impossible. In the words of David Grimaldi, an entomologist who specializes in fossil insects, if one were searching for dinosaur blood, an amberized fossil insect would be an imaginative place to look for it. But then the real difficulties would arise. You're in your laboratory, examining a piece of amber. If you hold the amber up to the light, you can plainly see inside it a mosquito. This mosquito is 85 million years old. Now what? In the movie of Jurassic Park, there's a scene where a close-up is shown of a little hole being drilled into the amber. A needle is inserted, and a syringe draws out something from the insect's stomach. Easy, but maybe not such a good idea. Any piece of amber, no matter how pure and translucent it looks, contains all kinds of microscopic bits and pieces of the past. If you drill a hole through the amber and insert a needle, you're going to pick up DNA from any number of life forms that have nothing to do with dinosaurs. Not to mention that the insect itself has its own DNA, and if you drill a hole to get to the insect's stomach, you can't help going through some of the insect's skin and muscle. Sawing the amber in half is a better plan, but you have to plan the job carefully. Once that amber casing is cracked apart, the insect is exposed to the modern world and to all the bits of DNA that are floating around in the air. Working slowly, you cut around the circumference of the nugget of amber using a small circular saw on the end of a jeweler's drill. You're careful to stop short of the mosquito itself. At this point, you can wash it liberally with alcohol to get rid of any DNA-containing material from whatever else was in the amber. Now you're ready to crack the amber open. But you need to do this in a place where all possible DNA contamination has been thoroughly eliminated. Here's how it's done. Ultraviolet light messes up the structure of DNA, altering the form of some of the DNA molecule's chemical components. That's why UV and sunlight is bad for the skin. It can alter DNA structure in your skin cells and trigger cancer. In your laboratory, you have a plexiglass glove box equipped with an ultraviolet light. You put all the equipment you need, instruments, chemicals, devices, into this box and switch the light on. Now you want to put your piece of amber in the box, but at this point you have to switch the ultraviolet light off because you don't want to destroy anything once you open the amber. The other feature of this box is what's called positive airflow. Clean, filtered air is forced into the box so that every time you open the box, air blows out. You don't want laboratory air getting sucked into the box after you've sterilized it. You work inside the box by putting your hands into gloves that are sealed to the front of it. Now you're ready to go. First, plunge the amber into super cold liquid nitrogen, which makes it more brittle. Now you simply crack the amber gently open, releasing the mosquito from its 85 million years of imprisonment. You want to extract as much of the insect as you can to study it further, but that's not so easy. You're going to have to break apart this perfectly preserved creature, but at least you can try, with the aid of a low-powered binocular microscope, to dismantle it carefully. You'd make sure there was a plentiful supply of sterilized tubes on hand, and you might well have stuck numbered labels on them all. That way, as you're dropping bits of the insect into the tubes, you can call out the number and describe what you're putting in there for an assistant to write down. So you pull off a leg, put it in a test tube, call out the number. Then a bit of wing, a fragment of skin, some hairs. And you do your best to scoop out the stomach contents, keeping them as separate from the rest of the mosquito as you can manage. Now what? For the moment, let's forget about dinosaurs and just think about how to get DNA from insect parts themselves. That's actually been done, and learning how to accomplish that will get us ready for the more difficult task of extracting dinosaur DNA. You work in a glove box just as you did when you were breaking the amber apart. You would use, in any one experiment, just a fraction of the insect remains you had recovered. You don't want to lose the whole lot if something goes wrong. The DNA you want will be inside cells, so you need literally to break the cells open. You can do this with something that's essentially a kind of soap or detergent. Cell membranes are made of fatty molecules called lipids, all bound together, and you use soap to disrupt the membrane just as soap dissolves grease spots on your clothes or dishes. You also add an enzyme that degrades proteins in the resulting mess, because some of those proteins can chew up DNA. Normally, the structure of a functioning cell keeps DNA away from these dangerous molecules, but once you have mashed the cell up, you need to move quickly to keep DNA out of harm's way. The mess you've created at this point contains all kinds of stuff besides DNA. The next step is to add phenol, an organic solvent, to the mixture. What this does is to separate the DNA away from other stuff in the mixture. The DNA molecules will end up floating around in the water from the soapy solution, while the phenol will dissolve most of the fatty molecules from the cell structure. To get as much DNA out of the sample as possible, you shake the test tube for a few minutes so that the contents are thoroughly mixed. After thorough mixing, you then spin the mixture in a centrifuge for a while, which helps the water and phenol separate. On top is the water, containing DNA and probably a few other protein bits and pieces, while below is the layer of phenol, which contains all the other cellular material and junk you don't want. The proteins tend to sit in a milky layer between the water and the phenol. Now, it's a fairly simple matter to suck the water layer from the top using a pipette, which is basically a fine plastic tube attached to a handheld device that can apply suction. Now you have, you hope, a drop of water containing DNA and not much else. You may remember we said that water damages DNA, but as long as you don't let the DNA sit in the water for too long, everything's okay. This method for separating out DNA is reasonably efficient, but, you won't be surprised to hear, not perfect. A few odds and ends from the insect cells will have got into the water layer along with the DNA, so there's one more purification step to be done. The movie has a realistic scene in which a scientist in the laboratories on Isla Nublar has just performed this final procedure and is showing off to the visitors a test tube with a weirdly glowing red fluorescent ring. The test tube contains a thick slurry of cesium chloride, which is closely related to plain old table salt, but quite a bit heavier and denser. You fill up a test tube with the salty slop, add the solution that contains the insect DNA, and spin the whole thing around very fast in a device called an ultracentrifuge. This machine can spin the test tube on the end of an arm that's going around as fast as 100,000 times a minute. Rapid spinning makes the cesium chloride solution denser at the bottom of the test tube than at the top. If this is all done right, then the small amount of DNA will gravitate to a place in the tube where its density matches the density of cesium chloride at that point, and all the other impurities will be somewhere else. Before adding the insect DNA sample to the test tube, you also remembered to add a dye that's designed to chemically attach to molecules of DNA. When you shine an ultraviolet light on the test tube that you've just taken out of the ultracentrifuge, you should see a red glow marking the layer where the DNA has ended up. You can pull out the DNA by sticking a fine needle through the side of the test tube. You remember to use a plastic one, didn't you? And sucking out the red layer. This is exactly what's shown in the movie when the technician proudly holds up a test tube with a glowing red ring. Still, there are a couple of problems with this scene as portrayed. First, you need a lot of DNA molecules to get a red ring that you could actually see. This is all very well if you're trying to extract billions of DNA molecules from a good-sized chunk of a living animal, but not if you're hunting for 1 or 2 molecules of dinosaur DNA. And second, the technology of ultracentrifuging test tubes of cesium chloride was on its way out when Michael Crichton wrote Jurassic Park, and was defunct the time the movie came out. Luckily, the second problem takes care of the first. There's a much better way to identify and extract DNA from scanty samples. It's also quite a bit easier. Let's go back to the point where we had just pulled the layer of water containing the DNA from the phenol layer. First, you add some ethanol, that's the official name for plain old alcohol, along with a little salt. DNA molecules will precipitate out in a mixture of water and ethanol, so when you add ethanol you should see the liquid take on a cloudy white appearance. That white precipitate is DNA. Next, you put your test tube into a standard issue lab centrifuge and spin it around for a while. This forces the DNA into the bottom of the test tube so you can pour off most of the water-ethanol mix and then leave the rest to evaporate. This gives you a dry sample of DNA. But not very much of it, perhaps just barely enough to see. Now comes the modern, almost magical step. You plunk the DNA into a test tube containing a specially prepared brew of chemicals, then stick the test tube into a slot in a hot plate that's controlled by a timer to raise and lower the temperature every couple of minutes. Then, 2 or 3 hours later, you come back. If all has gone well, there should be plenty of DNA in the test tube, millions of times more than you put in a few hours ago. Nice trick, huh? Are you sure you want to spoil the magic by finding out how it works? Well, okay. The trick lies mainly in the mix of chemicals that you added. To find out what those chemicals do, we need to learn a little more about what DNA is. DNA is a chain whose links, little chemical units in themselves, come in just 4 different types, designated by the letters A, C, T, and G. A DNA molecule is a string of those letters. In the case of human DNA, the chain is about 3 billion letters long. It's the specific sequence of A's, C's, T's, and G's that make each individual's DNA different from anyone else's. Nevertheless, every human being's DNA has to contain the same basic building instructions, so large fractions of the human DNA molecule are the same for every person on the planet. The variations control such differences as hair color, shape of nose, and so forth, but interestingly, there are many differences that seem to have no significance at all. Much of every person's DNA has no discernible function and can vary significantly from person to person. In the cells of your body, DNA takes the form of 2 such strings spiraling around each other to form the famous double helix. And there's a particular relationship between the 2 strings. Where there's an A on one chain, there must be a T on the other, and where there's a C on one, there's a G on the other. In all likelihood, the DNA you obtain from the ancient insect will have broken down, over the millions of years of its imprisonment, into smaller fragments. What you will have is a test tube containing bits and pieces of insect double helices. The machine that multiplies the insect DNA starts by heating the test tube to almost boiling point. At that temperature, the 2 strands in each double helix fall away from each other. Then the machine lowers the temperature. When the mixture is at about 72 degrees centigrade, the special ingredients in the mix you added start to kick in. Those ingredients include a plentiful supply of the individual bases, the A's, C's, T's, and G's, floating freely about. In addition, there's an enzyme called DNA polymerase, whose job is to ratchet along a single strand of DNA, reforming the double helix by fashioning the correct adjacent strand. In other words, if the polymerase is at a T, it will pick up one of the free-floating A's. If it's at a C, it will find a G and attach it. Where there was one double helix to start with, now there are two. The machine repeats this cycle over and over, doubling the amount of DNA at each step. 30 cycles, which the machine can complete in a matter of hours, will multiply the original amount of DNA by 1 billion times. This procedure is called the polymerase chain reaction, or PCR for short. It was invented in the mid-1980s by a biochemist named Kary Mullis, who won the Nobel Prize in 1994 for his invention. PCR became so useful to researchers that barely 5 years after its invention, you could buy from mail-order scientific equipment catalogs a simple machine that did the whole thing for you. So you go for a tea break while the PCR machine is humming quietly along. Eventually, you come back to the lab and pluck the test tube out of the machine. If all has gone well, there will be lots of copies of whatever DNA was in there originally. So now, on to the next step. Well, sad to say, it's not quite that easy. There was one additional fact about PCR that we forgot to mention. Polymerase doesn't work unless it's given a kick-start. To set the whole process rolling, the polymerase first has to latch onto the single strand so it can start ratcheting along, and to attach itself, it needs help. The other ingredient in the chemical mix that goes into the PCR machine is what's called a primer. This is a short single strand of DNA, perhaps just 15 or 20 bases long. After the machine has heated the test tube to make the DNA double helixes fall apart into single strands, it actually cools the solution all the way down to about 50 degrees centigrade, which is cool enough to let the primers attach themselves to the longer strands of separated DNA. Alright, you think, so we need to add primers. What's the big deal? The big deal is this. A primer will only be able to attach itself to the long pieces of DNA if it has an appropriate complementary sequence. There has to be a T opposite the A, a G opposite the C, and so on. If no sequence matches up to the primer can't attach, the polymerase can't get to work, and nothing will happen. If you know what primers to use, all is well. But to use PCR to multiply dinosaur DNA, you need primers that will latch onto dinosaur DNA sequences. Except that before you've found any dino DNA, you don't know any of its sequences, and so you can't make primers that you know will work. If you don't know what you're looking for, how can you find it? You're tagging along with Alan Grant, Ellie Sattler, Ian Malcolm and the others on their first visit to Jurassic Park. Henry Wu, the enthusiastic chief geneticist, is showing you around the lavishly equipped research laboratories of Isla Nublar. Wu is explaining how they get dinosaur DNA. First, they collect biting insects of the right age, preserved in amber. Second, with a fine syringe, they poke into the insects' stomachs to see what they can find. Only a few insects have any dinosaur blood in them, but for each dinosaur species, they only need one blood sample. Third, they carefully suck into the syringe as many red blood cells as they can find. It's easy to be sure that a red blood cell inside the insect doesn't belong to the insect itself, for the simple reason that insects don't have red blood cells. They have a kind of blood called hemolymph, but the molecules that carry oxygen around are not packaged up in little cells. Wu admits that the DNA they find this way might not be perfectly preserved, but says they've come up with methods to patch together pieces that have fallen apart and even to fill in occasional gaps. On to the next step, which is turning dino DNA into an actual dinosaur. Wait, there's a couple of little details we need to think some more about. First, there's a problem that Wu mentions, although only because he can explain why it's not really a problem after all. Human red blood cells, as it happens, don't contain a full complement of DNA. Red blood cells live for only about 100 hours, and their only purpose is to carry oxygen around in the bloodstream. Because the body has to keep churning out a steady supply of new red blood cells, it may be a matter of biological efficiency that the need to equip them with DNA was dispensed with at some point in evolutionary history. But dinosaurs, Wu wants you to believe, did have DNA in their red blood cells. This is not such an unreasonable idea. Dinosaurs are thought to be the ancestors of birds, and red blood cells from birds indeed contain DNA. So let's give Wu the benefit of the doubt and suppose there was DNA in red blood cells from dinosaurs. Unfortunately, there's another big problem that's been overlooked here. Why does an insect drink blood? To eat, of course. And what does an insect do after it's eaten? Why, it grabs the newspaper, turns on the TV, settles down on the sofa, and digests its dinner. Ah, digestion. In other words, the dinosaur blood gets dropped into the insect's stomach, and that's a tough place, awash with caustic chemicals and digestive enzymes. A simple red blood cell plunged into the harsh environment of an insect's digestive system just won't survive very long. Of course, we're assuming that the insect we're now looking at didn't survive very long after eating. But even if it got stuck on a resin-coated tree almost immediately after eating, it'll take a while for the resin's preservative chemicals to seep into the insect and kill it. And even after the insect is completely dead, the digestive reactions in its stomach won't instantly come to a halt. And just a few minutes inside an insect's stomach is enough to start wrecking the dinosaur's DNA. It's really not at all likely that an insect could have fed on a dinosaur, got instantly trapped in resin, and that preservation happened quickly enough that some of the dinosaur's blood remained undigested. Remember that all the DNA that's been extracted so far from organisms in amber came from the organisms themselves. At this point, you're better off abandoning the strategy of looking for dinosaur DNA inside an insect and try to come up with a more likely possibility. It was Jurassic Park's seminal notion that a mosquito biting a dinosaur could lead to the possible recreation of live dinosaurs. But once you start to think a little more closely about what's involved, it becomes almost impossible to imagine that the idea could work. The solution is to come up with another idea. You know that bits of DNA have actually been extracted from insects trapped in amber. And if a whole insect, with at least some DNA intact, can survive for that length of time, why not a piece of dinosaur? Dinosaurs must have fought, and in an especially vicious fight, a few chunks of dinosaur might have been sent flying through the air. Or perhaps some particularly messy eater tearing dinosaur meat apart would have left a few morsels lying around. One way or another, let's imagine that a piece of dinosaur could have got flung onto a suitable tree, swallowed up by resin, and then preserved in hardened amber to the present day. Assume that John Hammond has bought up every last piece of amber in the world, scoured every amber mine, and found what you need. Chunks of amber containing chunks of dinosaur. You can't tell directly whether a chunk of meat inside amber came from a dinosaur or a little Jurassic rodent. But that's also a problem with the original scenario. You couldn't be sure that blood inside an insect was dinosaur blood and not mouse blood. Any way you look at it, there's going to be a lot of trial and error in this part of the project. Now you're back in the laboratories on Isla Nublar, looking at morsels of dinosaur steak trapped in amber. That's progress, but your problems are far from over. Your preserved piece of meat may well have some DNA in it, just as amber-preserved insects and plant fragments are known to have DNA in them. But what's been extracted from insects and plant fragments, so far anyway, is no more than tiny snippets of DNA, not the animal's complete genetic code. The first example of DNA archaeology came from the lab of Alan Wilson and Russ Higuchi at the University of California at Berkeley. In 1983, they extracted DNA fragments from an extinct horse-like animal called the quagga, which used to live in South Africa but was hunted to extinction toward the end of the last century. Wilson and Higuchi took scrapings of dried muscle from a quagga pelt that had been preserved in salt in the Berlin Natural History Museum. They did succeed in finding some quagga DNA, and by comparing it to related segments of DNA from modern horses and zebras, they were able to figure out where the quaggas belonged in the equine family tree. They were more like zebras than horses, as it turned out. This was the first successful extraction and identification of DNA from an extinct animal. But none of the DNA segments that Wilson and Higuchi found were more than a few hundred letters or bases long. The complete DNA, known as the genome, for a typical mammal runs to something like 3 billion bases. A fragment of a few hundred bases is a very small piece indeed. On the other hand, the quagga pelt hadn't been preserved in an especially careful manner. Amber should do a better job of preservation than a drafty cabinet in a storage area of the Berlin Museum. There is still a question as to whether a substantial fraction of DNA can survive for millions of years, as opposed to a few thousand. The oldest DNA fragment ever recovered was from a 135-million-year-old beetle, and the fragment itself was 400 bases long. That length is typical of DNA fragments that have been retrieved from all manner of very old, amber-preserved insects and plants. There's nothing you can do about this part of the problem. You can only hope that nature has been kind. But this brings us back to the problem we ran into earlier. You can use PCR to search out and multiply individual fragments of DNA. But in the standard version of PCR, you have to supply primers. But if you're looking for DNA of an unknown type, then by definition, you don't know what primers to use. Once again, we're going to have to be inventive. You've carefully removed the alleged piece of dinosaur from its amber casing, using all the tricks you've learned to avoid contamination, and you've separated out the DNA. You've now got a little test tube containing microscopic quantities of dinosaur DNA. How do you get the DNA out? What you really need is a method that can pick up every last piece of DNA from your sample without any second guessing as to what the DNA might look like or where it might have come from. This strategy amounts to using PCR with random primers. If you don't know what kinds of primers to use, try all the ones you can think of. Any primer is a piece of DNA, a string made up of the letters A, C, T, and G. In a typical PCR application, such as DNA fingerprinting, the primer might be 20 bases or letters long. How many such primers can there be altogether? In each position of the 20-letter sequence, you can put any one of 4 letters. The number of primers consisting of 20 letters will be 4 multiplied by itself 20 times, which is a big number. About 1 trillion, in fact. For the dinosaur DNA problem, this is absolutely the wrong thing to try. If you threw a random 20-letter primer into one of your test tubes of dinosaur DNA, the chances are very slim indeed that you'd pick up anything at all. The answer might be to use shorter primers. For example, the number of possible DNA sequences with just 6 letters is a mere 4,096. But these primers, instead of not sticking often enough to unknown DNA, would stick too often. Although it's hard to predict exactly what would happen, these short primers would probably stick to the dino DNA, leaving only short gaps between them, perhaps 5 to 10 bases long. PCR would then amplify up just those stretches between the primers, which means you would get millions upon millions of extremely short fragments. That would be tough to handle. A reasonable compromise would be to use random primers that have a good chance of appearing once or twice in the hypothesized billion-letter specification of dinosaur DNA. It turns out that if you used random primers of 15 letters, there would be a little more than 1 billion different possibilities, which means that each one would have a good chance of occurring somewhere in your dinosaur DNA. This would tend to give you fragments a few hundred bases long, which is about the maximum you can expect anyway, given what we already know of actual DNA extraction from ancient insects and plants. But does this mean you'd have to do 1 billion separate PCR amplifications? Here's one of the few instances where something that seems daunting turns out to be easier than you might suspect. You can just throw all the billion plus 15 base primers into a single PCR tube with your dinosaur DNA and let the machine run. Even better, you can make those billion 15 base primers in about an hour. Once again, a little machine will do the job for you. You might be thinking that 1 billion different primers would be a lot of stuff to put in one little PCR tube, but it's not. Molecules are small. Even if you throw in 1,000 copies of each of the billion primers, you've still got only 1 trillion molecules. A sugar cube, by comparison, contains about 1 trillion individual molecules of sugar, so your package of DNA primers is only something like 1/trillionth of a sugar cube in size. No big deal. After PCR has done its work, you should be left with a test tube that contains all your original dinosaur DNA fragments multiplied many times over. You have lots of copies of all the dinosaur DNA fragments, but they're still all mixed up together, and you can't go any further until you've isolated each fragment so that you can analyze and identify them. This is where things begin to get a bit problematic. The best way to separate the DNA fragments would be to use some sort of standard cloning technique. Inside bacteria are little circles of DNA called plasmids. Scientists have learned to incorporate small fragments of foreign DNA into those plasmids and then to slip the plasmids back into the bacteria from which they came. E. coli is the bacterium of choice. The bacteria will grow happily with the bits of extra DNA inside their plasmids, and it's easy to keep large colonies of the bacteria alive. Now you've got a way of storing and multiplying all the bits of dinosaur DNA. Plasmids can't handle large DNA fragments, but for pieces of dinosaur DNA a few hundred bases long, they're ideal. The problems with this technique are logistical. You mix up the PCR dino DNA with bacteria so that each bit of DNA gets taken up by a bacterium. Then you smear the bacteria out on a plastic plate covered with a nutritious jelly, encouraging each one to grow into a colony of identical little bacteria, all carrying the same bit of dino DNA. You might be wondering why we needed to do PCR on the original dinosaur sample if the bacteria cloning method also takes individual bits of DNA and multiplies them up. The answer is that cloning is not 100% reliable, because you can't be certain that each DNA fragment in the original sample will be picked up by a plasmid, so it's a good idea to use PCR initially. Let's imagine that you end up with 100 million tiny fragments of DNA, each one tucked inside a bacterium that you're going to grow into a colony. If each little colony needs a square centimetre of space to grow in, you're going to need 100 million square centimetres of goo-covered glass. That area, regrettably, is a space 100 metres on a side, comparable to a couple of football fields. Of course, you'll have lots of little plates all stacked up separately, but you're still going to need a big lab. When you've grown the colonies, you'll need to scrape them off into individual test tubes for the next step. To store 100 million little plastic tubes in some kind of automated warehouse, you're going to need a space the size of a large department store. And think about time and manpower. Every time you have to perform a routine operation on your samples, you've got to do it 100 million times over. It would take 3 people a whole year just to do something with every test tube that takes a second per tube. Oh well. At this point, let's just assume that with all of John Hammond's resources, you'll somehow get the job done. At the end of it, there'll be 100 million little test tubes containing between them remnants of dinosaur DNA. Now, you just have to see what's in all those test tubes. The first order of business is to find out the sequence of A's, C's, G's, and T's that make up every last one of those fragments. The task of sequencing pieces of DNA is one that, like so much else you've been doing, has become largely standard and automated in a modern biology laboratory. Here's how you sequence a DNA fragment. You put it in a machine called a sequencer. In a matter of hours, the machine can sequence a piece of DNA about 1,000 bases long. Longer pieces are more troublesome, but since our dinosaur DNA fragments are highly unlikely to be more than a few hundred bases long, each one can be sequenced directly. A good sequencing machine, costing around $125,000, can sequence as many as 64 strands of DNA at once. Let's see. You've got 100 million test tubes, and you can do 64 sequences at once. That means a little under 2 million sequencer runs. Each run takes an afternoon, allowing mornings for cleanup and preparation, so you'll need about 2 million sequencer days. That's 2,000 sequencers, costing a total of $250 million, operating continuously for about 3 years. Not to mention all the technical staff you'll need, lab space, chemical supplies, cleanup crews, electricity bills. Even so, the task seems tedious but not infinite, expensive but not prohibitive. At this point, you've isolated a whole stack of little fragments of dinosaur DNA, and you want to put them together to make the complete DNA of a dinosaur, its genome. Trouble is, you don't know what the genome looks like in the first place, so it's going to be difficult to figure out how to put all the pieces together in the right order. In an enterprise more or less the opposite of the one we're faced with, known as the Human Genome Project, scientists around the world are embarked on an effort to figure out the complete sequence of the genome of a human being. The Human Genome Project is a basically straightforward but enormously time-consuming and costly task. The main difficulty is simply that modern sequencing technology can only handle 1,000 bases or so at a time. If the human genome is something like 3 billion bases long, then you're looking at cutting it into 1 million or so small pieces, all of which have to be sequenced individually. That in itself is a daunting task, but there's a more subtle difficulty. If you cut a single genome into 1 million little bits, how do you keep track of where all the pieces belong? It's not much good having the sequences of 1 million pieces if you don't know how to connect them all again. On the other hand, there's no way to do the sequencing without cutting the genome into smaller and more manageable pieces. Sequencing the entire human genome is a laborious process, as much of an exercise in bookkeeping and management as in science itself, and things don't get much simpler where dinosaurs are concerned. At this point, you can take a break from the DNA extraction and sequencing lab and all those tedious and messy test tube procedures. As you try to reassemble the dinosaur genome, what you mainly need is big computers. Fortunately, John Hammond supplied Isla Nublar with 3 of the biggest supercomputers his money could buy. With the sequences of all the DNA fragments safely stored in computer files, you can compare, sift, and manipulate them to your heart's content. The problem of trying to put together a number of pieces of dinosaur DNA in the right order to make a correct dinosaur genome is something that Henry Wu is well aware of. As he's showing his visitors a seemingly random string of the letters A, C, G, and T displayed on a computer screen, he proudly declares, "Here you see the actual structure of a small fragment of dinosaur DNA." As he's displaying the DNA sequence, Wu also points out that there's a glitch in the sequence, a little section of 5 bases or so that appears to be missing or unidentifiable. Now that raises a question. What Wu has actually put up on the computer screen is one long sequence of DNA, followed by a small break or gap, followed by another sequence of DNA. He implies that somehow he knows these 2 sequences match up. But how does he know that? Conceivably, he may have been trying to sequence a long stretch of DNA, and the sequencing machine figured it all out except for a little bit in the middle. However, that would assume you've been able to find long fragments of intact DNA, and we've come to the conclusion that isn't likely at all. The actual problem you face is that you have a great many sequences, all a few hundred bases long at most, and you have absolutely no way of figuring out the correct way of putting them all together again. A single set of DNA fragments won't do then. But when you find a way of retrieving DNA from amber, what you're going to get is not fragments of DNA from just one cell, but a whole mixture of fragments from several cells with the same genome. This makes life a lot easier, because some of those fragments will overlap in places, giving you a way of joining pieces together. This process of building up contiguous segments of DNA, contigs, scientists call them for short, is a routine procedure in the Human Genome Project and in many other smaller-scale researches. This technique is strictly an exercise in looking for patterns and matching them up. You don't need to know anything about what the DNA actually means in order to put all the pieces together. It's painstaking work, to be sure. If the dinosaur genome is a few billion bases long, and if it's broken up into pieces that are typically 500 bases long, then you're going to be faced with an awful lot of tiny scraps. A couple of supercomputers should be able to polish the job off pretty quickly, however. So far, we've made it sound as if you could load all your sequenced DNA fragments into a supercomputer, set the wheels whirring, and after a day or 2 the dinosaur genome would pop out, reconstructed perfectly. But would this really work? Sadly, no. There are a few complications we haven't mentioned. Like the human genome, the dinosaur genome was presumably divided up into chromosomes. We have no way of knowing how many chromosomes there should be when we put DNA back together again. Humans have 23 pairs of chromosomes, but gorillas have 21 and chimpanzees have 24, even though all of us are genetically very similar. Among other animals, some kinds of salamanders have hundreds of chromosomes, while the muntjac, a small Asian deer, has but one pair. That isn't the whole chromosome story, because we can assume that dinosaurs, like humans and indeed all mammals, reptiles and birds, would have possessed two sets of chromosomes, one from each of their parents. To fully reconstruct your dinosaur, you would have to have both sets of chromosomes in all their intricate detail. The division of the dinosaur genome into chromosomes presents the supercomputer with a new difficulty. But there may be a way around this problem. It turns out that chromosomes always end with long, repetitive sequences called telomeres. The biochemical machinery within a cell, just like PCR, needs a primer to get it started. Telomeres, in effect, provide a standard piece of DNA that the copying machinery can latch onto to make new copies of the chromosomes. For the purposes of figuring out dinosaur chromosomes, you would have to tell the computer to look for lengths of repetitive sequences, which might be telomeres. Or they might not be. There are lots of places in any genome where you can find simple sequences repeated many, many times. These apparently dull sections of the genome, which are called microsatellites, can be hundreds or even thousands of bases long. There are long stretches of any genome that have been given the derisory name junk DNA because they seem to serve no obvious purpose. Another vexing difficulty is that the dinosaur genome, trapped in amber for millions of years, may not fall apart entirely at random. Certain sequences seem to be more fragile than others, and if there were a number of places in the genome that always broke first, your reconstruction attempts could never bridge those gaps. But apart from all this, the real problem is in assuming that you have all the pieces of a dinosaur genome in the first place. Judging by what's been found with 30-million-year-old insects, you'll be lucky to retrieve just a handful of small fragments. And if there are gaps in the recovered dinosaur DNA, you come to a dead stop. There's no way of knowing whether 5 bases are missing or 5 million. Wu's explanation of putting the dinosaur genome together, though it has some plausible elements, is in the end highly implausible. Unless the preservation of DNA pieces is perfect, it just won't work. We're being a little unfair here. Wu has another trick he uses to repair the DNA fragments. As the little cartoon in the Visitors Center notes, there were times when the Jurassic Park scientists were faced with gaps in the DNA that they couldn't bridge. In those cases, Wu says, they resorted to patching up the genome with bits of DNA from other species, frogs to be precise. This gets the park in trouble later on when it appears that the frog DNA has caused the all-female population to produce a few male dinos, but we'll get to that later on. For the moment, let's think some more about using DNA from one species to repair the DNA of another. This might sound at first like a pretty bad idea. After all, the reason one species of animal looks different from another is fundamentally that they are made from different genomes. On the other hand, the DNA of closely related species is, by the same argument, also closely related. Think about humans and chimps again. Although a person and a monkey are obviously different animals, they also have a lot in common. That includes not just general appearance, but internal organs, bone structure, blood chemistry, immune responses, and so on. All of these systems are built from instructions in the respective creature's DNA. To understand this better, we're finally going to have to learn a bit more about how the genetic code embodied in DNA actually works. The full story on this is something scientists have not yet figured out, but a good deal has become clear since the discovery of DNA structure just under 50 years ago. Take blood as an example. The molecule that carries oxygen through your bloodstream is called hemoglobin. Hemoglobin is a protein, one of several tens of thousands of proteins that are essential to the structure and functioning of a living body. To be precise, hemoglobin is made of 2 copies of 2 different proteins hooked up in a particular way and centered around a molecular assembly that performs the important task of ferrying oxygen around. The proteins themselves are made of smaller chemical units, called amino acids, and the instructions for putting the right amino acids together in the right way are coded into your DNA. All proteins in your body are strings of amino acids, which come in 20 different types. For hemoglobin, the instructions for putting the roughly 600 amino acids together in the right way reside somewhere in your genome as two individual genes for the two proteins that make up hemoglobin. A gene is a stretch of DNA that carries the code for a string of amino acids. If a gene is just a stretch of DNA that has the instructions for making one particular protein, then the genome must be a long list of genes, one after the other, right? Unfortunately, it turns out to be a lot more complicated than that. Stretches of DNA that carry the instructions for a sequence of amino acids are called coding DNA. The rest is called, obviously, non-coding DNA. You might be surprised to learn that only about 5% of the whole genome, in humans at any rate, is coding DNA. The remainder, a full 95%, therefore doesn't carry instructions for making proteins. So what's it all for? The truthful answer is that no one knows the whole story. Some non-coding DNA seems to genuinely deserve the name junk. It just doesn't do anything at all. There are other large sections of non-coding DNA whose purpose is only now beginning to be understood. Other sections of non-coding DNA are purely functional, acting as switches. For instance, a piece of DNA called a promoter, found at the front end of certain genes, sets off a chain of events that ultimately results in the instructions within the gene being turned into the protein it codes for. Other sections of non-coding DNA may serve architectural roles, that is, they hold DNA together in a particular shape and form. There are also pieces of non-coding DNA within genes, not just between them. These interpolated sections are called introns, and they may well occupy much more of the sequence in a gene than the coding sections themselves. When a gene is read to make a protein, the introns are in essence passed over and snipped out. You can see that there's more to reconstructing a gene correctly than just coming up with the right sequence of coding DNA to make the protein you want. Think about hemoglobin again. Every person contains hemoglobin genes, but those genes are not identical from one person to another. There are also small variations in hemoglobin itself from person to person. Some of the 600 amino acids in hemoglobin can be substituted by others without any appreciable change of the performance of the hemoglobin itself. Other substitutions, however, produce hemoglobin that simply doesn't work. Then there can be differences in the huge amount of non-coding DNA whose consequences are quite unpredictable. This range of possible differences, both serious and inconsequential, makes the job of repairing dinosaur DNA very hard. If you were trying to reconstruct a human genome by patching it with pieces from a chimp genome, you would probably be able to do a pretty good job. Henry Wu, on the other hand, is trying to fix up dinosaur DNA with bits of frog DNA. The choice of frogs is mystifying. If you were trying to do this at all, your best bet would be to look at bird genomes, because birds are thought to be the nearest living relatives of dinosaurs. But modern birds are certainly not close relatives of the dinosaurs. Because dinosaurs died out so long ago, and because birds have evolved so much in the meantime, it's not at all likely you could find, tucked into the genome of some modern bird, the bits and pieces of genes you would need to patch up incomplete dinosaur genomes. Then again, you don't really have any other option but to look. You've patched together, as best you can, all the fragments of dinosaur DNA you've managed to collect. At this point, you go to the birds. That is, you pick a variety of birds, including some primitive ones such as emus and ostriches. You then sequence completely the genomes of as many as possible. Bear in mind that this will cost billions of dollars and keep many researchers occupied full-time for about 2 decades. When you've done all this, you compare the dinosaur DNA fragments with all the bird sequences that you've now obtained. What you're looking for is similar or perhaps even identical sequences. If it's a sufficiently long sequence to be essentially unique, you use the bird genomes both as guides to where the corresponding bits of dinosaur genes might belong on the dinosaur genome, as well as a model for filling in missing pieces. If you can recognize a single dinosaur gene as it emerges from the reconstruction process, you might also want to start thinking about what this particular gene does. You might try inserting it into the fertilized egg cell of a living animal to see what, if any, physiological effect it will have. Because of the interconnected evolutionary history of life on Earth, it's not unusual to find similar genes performing similar functions in, say, humans and mice and even fruit flies. Scientists have snipped an important gene out of a developing fruit fly egg, making it unable to grow into a fly, and then substituted a comparable gene taken from a mouse, which allows the egg to develop properly. Even though the mouse and the fruit fly genes are not identical, their structure and function are similar enough to be interchangeable. Scientists have also taken mouse genes known to be involved in eye growth and inserted them at random places in a fruit fly's genome, causing the fruit fly to sprout authentic-looking eye structures on its wings or legs or other odd places. If a foreign gene is taken up into the genome of another species, there's no guarantee it will do anything at all. But if you're lucky, the dinosaur gene will not only become incorporated into the developing mouse or fruit fly embryo, but will also have some interesting effect or another. Adding a single dino gene won't cause the mouse to grow scales on its back or develop huge, powerful hind limbs with fearsome claws. Those kinds of features are not controlled by single genes, but by whole cascades of genes and proteins interacting and feeding back on each other through the complex course of an embryo's growth. But it's not unreasonable to hope that at least a few dinosaur genes could have some significant effect on the developing host, perhaps as obvious as an alteration in the texture of a mouse's fur, or as subtle as a minor change in blood chemistry. Experiments of this sort could conceivably teach you not only about the function of those particular dinosaur genes, but also a little about dinosaur physiology and biology. But your principal task, of course, is to create the entire dinosaur genome, and your method has been to identify individual dinosaur genes by placing the dino DNA fragments alongside similar sequences within known bird genomes. The end result, though, will be a sort of fake genome, based on some sort of average or extrapolated version of all the bird genomes, with exact pieces of dinosaur DNA attached wherever you are sure of their placement. If this sounds like a wildly hit-or-miss procedure, it is. You can only hope that you get enough dinosaur DNA to allow reasonable coverage of the whole genome, because where you don't have dino DNA, you're going to have to fill in the gaps with guesswork from the bird DNA sequences. As long as you're willing to make enough guesses, you can come up with a sort of wannabe dinosaur genome. Whether this will correspond to anything like a real dinosaur, or indeed any kind of living creature at all, is something you have to pray for. The less dinosaur DNA you've been able to find, the more your wannabe dinosaur genome is going to look like the genome of a bird with a few dinosaur bits in it, rather than a dinosaur with a few bird sections. If you're going to indulge in extravagant thinking, here's something even more far-fetched than using bird genomes to help you construct a dinosaur genome. Why not try breeding birds in a highly selective manner with the eventual aim of creating dinosaurs? That is, you would breed birds so as to bring out, bit by bit, the dinosaur characteristics that still lurk within their genetic constitutions. It would be a bit like breeding dogs, as people have done for centuries, to amplify whatever qualities of speed or size or intelligence the breeder requires. On their own, dinosaurs bred one generation after another, and as the eons went by, those generations changed imperceptibly into whole new species. In one particular lineage, that process led ultimately to what we now recognize as birds. There must have been a series of increasingly bird-like dinosaurs, culminating at some essentially arbitrary point in creatures we would tend to call birds rather than dinosaurs. Every step in this long chain of breeding and reproduction consists of two creatures combining their genomes to make offspring. To create dinosaurs from birds, we would need to perform this evolutionary sequence in reverse, finding suitable pairs of offspring to breed so as to recreate genetically plausible parents. The idea springs from a suggestion made by the British evolutionary biologist Richard Dawkins in his book The Blind Watchmaker, to the effect that it might be possible to resurrect the dodo by selectively breeding pigeons, say, for increasingly dodo-like characteristics. But even transforming pigeons into dodos, which are, relatively speaking, close cousins, would be a huge task. Many people over the years have devoted their lives to breeding faster racehorses or redder tomatoes or bigger roses or sleeker greyhounds, but it can take enormous amounts of time and luck to come up even with rather small changes such as these. If you're breeding the conventional way, by getting two plants or two animals together and letting nature take its course, there's always a large element of randomness involved. You might take two super-fast greyhounds, a male and a female, breed them, and be disappointed to discover you got only average offspring because the parents' two sets of genes didn't get mixed up in the ideal manner to produce speedy youngsters. To breed dinosaurs from birds this way would be an altogether formidable job. As evolution proceeds in the forward direction, each new generation has a host of tiny changes compared to the parents' generation. Some of those changes allow the animals to prosper, while others may be disadvantageous. Although the survival of the fittest seems to impose a direction on evolution, in that dinosaurs gradually changed into birds, the direction is apparent only in retrospect, and wasn't built into the system at the beginning. The path that led from dinosaurs to birds was one path, selected by evolutionary pressures, from countless possible paths. To reverse evolution and go back the other way, you need to retrace that single path accurately, avoiding the myriad sidetracks and dead ends. Trying to breed two birds to produce a slightly more dinosaur-like ancestor would be like expecting two humans to produce a Neanderthal offspring. Even if it were a remote mathematical possibility, which is itself debatable, the chances of it happening are astronomically tiny. You'd be looking for just one combination out of a trillion possible combinations that might result when two birds mix their genes. The trouble with trying to breed birds into dinosaurs by making evolution run backwards is that you would be trying to retrace all the myriad intermediate steps between dinosaurs and birds without knowing what those intermediates looked like. Even if you did manage to produce some weird-looking offspring, how could you tell whether it was closer to a dinosaur than the birds you created it from, or further away? And there's one last problem. Birds descended, so it's thought, from one particular branch of the dinosaurs, creatures that started hopping and leaping and gliding short distances on feathery extensions of their limbs. The split-off of birds from dinosaurs happened long before the dinosaurs themselves became extinct, so there's a whole variety of other dinosaurs that you couldn't hope to recreate by breeding birds. Those unattainable dinosaurs are the ones that became truly extinct and left no living legacy. You could aim to produce dinosaurs corresponding to the direct ancestors of birds, but not these others. And you want to know which are the particular dinosaurs you could produce by breeding birds backwards? Wouldn't you know it? It happens that the direct ancestors of birds were the raptors, the villains of Jurassic Park. At the end of Jurassic Park, the island is destroyed and the dinosaurs are gone. How to produce a sequel? No problem. It turns out that there was a second island, the mysterious Site B, where Hammond's crew actually created and bred the dinosaurs. On Isla Sorna, the location of Site B, Malcolm and his new set of colleagues come across a whole new setup with laboratories the size of 2 football fields. This place is Hammond's dirty little secret, Malcolm explains, the place where all the experimentation and trial and error and mistakes went on behind the scenes. There were 1,000 failed dinosaur births for every animal that was actually born. And even after a successful birth, there was a staggering rate of disease and infant mortality. So now, you think, the laboratories of Site B, remaining much as they were when they were abandoned 5 years before, will finally reveal the biggest secret of Jurassic Park. How do you turn DNA into a dinosaur? Except, unfortunately, you still don't get much of an answer. And for a good reason. Although DNA is indeed the complete genetic blueprint for the animal it belongs to, it doesn't turn into that animal of its own accord. Along with the instructions for building an animal, the DNA, you need the right set of tools and materials. You have dinosaur DNA and nothing else. Or, to be more precise, you've stored in your computer a list of all the bases that make up a complete dinosaur genome, with the whole thing divided up into the correct number of chromosomes. It's certainly possible to turn a genome such as this into a whole animal, because that's basically what a chicken does with an egg. The problem is, no one really knows how this trick is accomplished. The egg contains a lot more than just DNA. It's a complex molecular assembly that can read the genetic instructions and make a chicken. Your job is to reproduce a dinosaur's egg, or at least provide a close enough mimic of it that your hard-won dinosaur DNA can be set to work. When you think of an egg, the first thing that comes to mind is probably one of those oval, hard-shelled things with a squishy yolk and white inside. To a scientist, the word egg has a more specific meaning. It's a single cell, produced by a female, that needs to be fertilized by male sperm in order to grow into a new creature. If dinosaurs had been mammals, you would be faced with an insurmountable problem. You would have to get a dinosaur embryo to grow inside the womb of some other similar animal, of which no such thing exists. But dinosaurs were egg layers, and that makes the problem a little easier. Impossible by present-day standards, but perhaps not entirely unimaginable. The fact that dinosaurs laid eggs isn't speculation. Fossil hunters have found a variety of dinosaur nest sites containing unbroken eggs, egg fragments, and infant and juvenile dinosaurs. Many fossil eggs are about watermelon size, not too different from the size of an ostrich egg today, for example. The yolk and white of an egg are generally unremarkable mixtures of proteins and fats, and probably wouldn't be too difficult to fake in the laboratory. That's what Henry Wu seems to have done, as he explains that they use a synthetic plastic material for the eggshells themselves. But that doesn't explain the truly difficult part of the problem, which is to make the egg cell. You can't just throw DNA from one creature into the egg of another and expect something to start growing. Think about what happens as an egg develops into an embryo. Once the egg cell gets fertilized by a sperm, it divides into 2, then 4, then 8 cells in a little cluster. If this simple pattern of growth were to continue, all you'd get is a bigger and bigger blob of identical squishy cells. For a real creature to develop, different cells have to start growing in different ways. Some become skin or muscle, others become nerves or liver or blood. Just how that happens, no one really knows. To grow dinosaurs, you're going to need yet more guesswork and luck. How are you going to install your hard-won dinosaur genome into any kind of cell, let alone a cell that might grow into a dinosaur? In the cells of anything more complicated than bacteria, DNA resides in a central structure called the nucleus. Even within the nucleus, DNA isn't just floating freely around. It's wound up and stored, to use an image due to the scientist Christopher Wills, like great lengths of rope stowed away in the holds of a ship. The entire human genome, if you could unwrap it completely and stretch it out, would be about 3 feet long. And in almost every one of the 100 trillion cells of your body, you have 2 of these 3 feet long molecules. The precise way the genome is stored may have significant effects on the things DNA can do in any one kind of cell. Think about 2 different types of cell in your own body. Take liver and skin, for example. Both liver and skin cells contain your complete genome, the whole DNA. But in liver cells, only the genes relevant to your liver are read, while in skin cells, only the genes needed to make other skin cells are used. In other words, if the genome is a huge encyclopedia, then in your liver cells it's open only at the pages marked liver. It seems likely that chromosome structure has something to do with how genes are read in different kinds of cells. Similarly, it's also quite possible that the way the chromosomes are wrapped up has some influence on the way genes work together. In other words, if chimps were suddenly all wiped out except for their DNA sequence, there's no guarantee you could recreate real live chimps by wrapping their DNA onto the structures of a human cell. If dinosaur DNA is to function correctly inside a fake dinosaur egg, it must be correctly packaged in such a state that the biochemical machinery of the egg can activate the necessary steps in the right order. We'll just have to suppose that biologists and geneticists will begin to understand how you might prepare a genome so that it can be installed within an egg's nucleus. In Jurassic Park, it seems that Henry Wu inserted complete dinosaur DNA into unfertilized egg cells taken from crocodiles, then inserted the modified cells into fake eggs. But by the time a fertilized egg is laid, it already contains an embryo that has developed too much for you to be able to turn it into a dinosaur. Its cells are starting to take on distinct functions. You may be able to persuade a crocodile to lay unfertilized eggs, just as chickens are made to lay unfertilized eggs for the supermarket, but an unfertilized egg lacks the biochemical structures into which you could insert dinosaur DNA. You have a much more difficult task. You have to remove crocodile DNA from the crocodile egg cell, or at least inactivate it, replace it with dinosaur DNA instead, and then persuade the egg that it's actually been fertilized so that it starts growing. But then what do? Wu implies that the yolk of an egg is nothing but a nutritious glop which they can synthesize in the lab to provide an environment for the modified egg cell to grow in. But it's not that simple. An egg cell needs to be properly implanted inside an egg with a yolk containing a variety of maternally provided ingredients essential to its proper development. Here's the best strategy we've been able to come up with. First, instead of using crocodiles as your source of eggs, you would use ostriches. Dinosaurs are closer in evolutionary terms to ostriches than they are to crocodiles, so dinosaur DNA might conceivably be a little more compatible with ostrich egg cells than it would be with crocodile egg cells. More important, since you will be attempting to grow dinosaurs inside them, ostrich eggs are bigger than crocodile eggs. Oh, and you'll have to explain to John Hammond that you're going to need an ostrich farm. Following Wu's example with the crocodiles, you would begin by extracting egg cells from an adult female ostrich, then replacing their DNA with dinosaur DNA. But rather than trying to make a completely artificial egg, you would then reinsert the modified eggs back into the ostrich's reproductive system, in the hope that the ostrich will then take up the lab-fertilized egg cell, pack it up in an egg with the requisite yolk and white, form a shell around it, and lay it. You may have to give the ostrich some sort of hormonal drugs to kick its egg-making system into action with the modified egg, but these are in a very general sense the same kind of problems that physicians tackle when they perform in vitro fertilization for humans. The advantage of this procedure is that you get the ostrich itself to do all the mysterious things that need to be done for an egg cell to start turning into an embryo. Once an egg has been laid, you would take it from the ostrich and into the laboratory to see if the experiment has been a success. Although no one knows how to fool an ostrich egg into making a dinosaur today, however, this doesn't mean no one will ever know. Understanding embryo development is one of the consuming problems of biology today. We simply have to assume that one day the process will be mapped out and fully understood. At that point, we can return to the Jurassic Park story, with little dinosaur embryos growing inside their laboratory eggs. After Henry Wu has explained their procedure for reconstructing dinosaur DNA, Alan Grant, the fossil expert, asks him how they know what kind of dinosaur it encodes. "We have two procedures," replies Wu. The first is something he calls phylogenetic analysis. That's basically the idea of trying to figure out a family tree of evolutionary relationships for genomes. If you know enough about the genomes of birds and other modern creatures, you can guess where, in this big picture, a dinosaur genome would fit. Scientists are beginning to understand such relationships. But for animals as disparate as extinct dinosaurs and modern birds, it's highly unlikely you could get enough information this way to examine a reconstructed genome and say what dinosaur species it came from. So you'd have to go to Wu's second procedure, which is, grow it and find out what it is. You've struggled to assemble a complete dinosaur genome, or at least what you hope is a complete dinosaur genome, from tiny scraps of DNA, and you fiddled and tinkered with ostrich eggs to persuade them to take up this alien DNA as if it were their own. Now all you have to do is stand back, keep the egg warm, make sure to roll it around regularly, and wait for a little dinosaur to poke out of the ostrich egg's tough shell. So you hope. Whether things actually run quite so smoothly is another matter. So that you can see what's happening, you snip out a window in the eggshell using fine surgical scissors. You cover the window with a piece of transparent plastic so that you should be able to see the contents. When a bird embryo begins to grow, it starts as a disc-like layer of cells nestling on the surface of the yolk. Needless to say, you don't know in advance what a primitive dinosaur embryo ought to look like, but it too would presumably begin as some sort of structure lying on the yolk. However, unless you were lucky enough to put the window into the eggshell just where this little cluster happened to be, there's no guarantee you could see it. It's big enough to see with a moderately powered microscope, but it might be on the other side of the yolk. With chicken eggs, you can often see the outline of an embryo once it's a few days or a week old by holding the egg up to the light. But an ostrich egg has a much thicker shell, and the yolk itself is larger and more opaque. Chances are you won't see anything, even if the experiment has worked. On the other hand, if a week or two goes by and you still don't see anything, you will have to conclude you've got a dud. You must be prepared, as the technicians of Site B were, for much more disappointment than success. But let's be optimistic. After you've looked at 1,000 or 10,000 ostrich eggs, you at last find one in which something seems to be growing. And let's be additionally optimistic, and suppose that you were fortunate enough to put a plastic window in the eggshell at the right spot, so that when you peer inside, you can see a tantalizing cluster of cells on the surface of the yolk. If you get to the point where an embryo is showing signs of a backbone and gut, of limbs and a head, you can be hopeful that some reasonable set of instructions from the genome is pushing the growing cluster of cells in a particular direction. But you still can't be sure whether you're going to get a dinosaur, or a dinosaur with some ostrich bits in it, or a mixture of different kinds of dinosaur, perhaps because you accidentally got DNA from more than one dinosaur into the set of fragments you painstakingly put together. Or even a dinosaur with bits of chicken and emu and eagle and sparrow from all the other bits of additional DNA you had to use to patch the whole thing together. The embryo continues to grow, you hope, and the yolk diminishes as the nutrients it provides are used up. You begin to anticipate the moment when it's big enough to start breaking out of its birthplace. All this time, of course, you've been taking special care of the egg itself. You can't just leave it lying around on a benchtop in your lab unguarded. In Wu's laboratory, eggs are set out on large trays in a room that's kept at 99 degrees Fahrenheit and high humidity, conditions meant to mimic the Jurassic climate that obtained in dinosaur stamping grounds. The factor that would most likely be of genuine importance here is temperature. Obviously, cold eggs don't develop, and hot eggs cook. You want the temperature to be just right. But there might be more to it than that. For some lizards and turtles, egg temperature controls the sex of the animal that hatches. An egg that's kept cool will develop into a girl, while one that's kept a little warmer will be a boy. Henry Wu, as he explains at some length, is anxious to make sure that his dinosaurs all turn out to be females so that they can't breed. He neuters them with radiation later to make sure they're sterile, but he also controls the development of the egg with hormones. Although the division of the world into males and females is widespread among all kinds of birds, mammals, reptiles, and fishes, the way the division is accomplished is remarkably varied. Quite a few species of fish can change sex in both directions on a number of occasions during their lifetimes. Some fish have the ability to generate male or female organs at any time during their lives, prompted by an appropriate hormonal trigger. What about birds? They're the closest relatives to dinosaurs, so maybe their way of sex determination is the one the dinosaurs might have used. It turns out that they have a sort of mirror image of the usual mammalian system. In birds, it's the males who have 2 copies of the same sex-determining chromosome, while the females have 2 different kinds. If dinosaurs worked the same way, Henry Wu would have faced an additional problem in making females. If he has only 1 copy of a single genome, and it was from male, then doubling it up will make a male animal. But if the single chromosome was for a female, it probably wouldn't work at all. Then again, we can't be sure that dinosaurs would have worked the same way as birds. It's possible that Wu's method of making female dinosaurs by withholding the necessary male hormones from the embryos might work, but even that assumes he can figure out what is the appropriate combination of male and female hormones to use at certain critical times in embryo development. Still, there's one surefire way of making the dinosaurs sterile. Just wait a while and do what vets do to puppies and kittens. You might want to do that operation sooner rather than later with T-Rex. But if all the animals on Jurassic Park were surgically neutered, you'd lose one of the key points of the story. Because they've gone out of their way to make all the dinosaurs female, Hammond and Wu are astonished and disbelieving when they find that the creatures are reproducing. The explanation, as Alan Grant finally figures out, is that dinosaurs with bits of frog DNA in them can change sex. Females can become males, perhaps because of some external influence. Certain frogs and amphibians are able to change sex, and so the idea is that dinosaurs with frog DNA would have acquired the same ability. This is hard to believe for several reasons. First, out of all the DNA of a frog, only some relatively small fraction has anything to do with gender determination. It would be a huge coincidence if the bit that Wu used just happened to be a piece that played a role in the frog's sex change habits. Second, what controls the development of sex is not just one gene, but a whole complex interaction of many genes. Nor would all the relevant genes of the frog be confined to one place in its genome. There could be bits and pieces all over. Third, even if Wu had, by some astronomical mischance, put all the right pieces of frog DNA into a dinosaur, there's no reason at all to think that the specific actions of those genes would have anything like the same effect in dinosaurs. Snipping out one set of genes from a dinosaur and replacing them with some very approximately equivalent set of genes from a frog will almost certainly result in a genome that simply won't work at all. The bottom line here is that there will be many things you can't figure out except by trial and error. You have to learn how to insert DNA into an ostrich egg, what sort of temperature and conditions are best for making the embryo grow, how long you have to wait before anything pops out of the shell, what signs to look for that something is going wrong. You've seen that the embryo was starting to grow and taking on a shape, and then you waited, and you waited, and then you waited some more. Chickens take 22 days to break out of the egg after they've been fertilized. Ostriches and emus take about 3 months. You don't know how long your dinosaur will take because, for one thing, you don't know what sort of dinosaur it is. If you have one of the giant dinosaurs growing inside an ostrich egg, you figure the thing is bound to bust out sooner or later. But what if the dinosaur embryo is a little Procompsognathus, one of the chicken-sized scavengers that scuttle about Jurassic Park cleaning up the mess? If it's about the size of a chicken, perhaps it needs a chicken-sized egg too. There'd be many failures before you had the first success. But one day, after you've figured out what size egg to use, you will achieve that wonderful moment when the egg begins to crack and a beak, or a snout, or a claw begins to poke out. At last, the egg breaks fully apart and a little dinosaur, about a foot from head to toe perhaps, struggles to its feet. This is the first dinosaur to be born for 65 million years. It's looking out on a world its parents never saw, a strange world with racks of test tubes on the horizon and odd-looking pink-fleshed creatures staring at it in wonderment. But the newborn dinosaur doesn't know that this world is not the one it was meant to be born in. All it knows is that it's cold and hungry. It starts squealing and waits to be fed. Baby velociraptors seem to be pretty darn cute. In the hatchery, a newborn raptor snuggles up to Tim, Hammond's grandson. Later, when the park is coming apart at the seams, Tim and Alex are back in the visitors' center and come across the same little infant, starving and desperate for affection. But now, Tim and Alex are being stalked by adult raptors who are not nearly as cuddlesome. Attempting to distract the raptors, Tim flings the baby toward them, hoping they will give up the pursuit to take care of it. Instead, they devour it, fighting among themselves for the scraps. But if the grown-up raptors are so fierce and smart and wicked, would they really have been such adorable cuties when they first emerged from the egg? Newborn alligators and crocodiles, for example, come equipped with a full set of teeth and a bad attitude. You'd be pretty unwise when the first dinosaurs are born to play with them as if they were chicks or bunny rabbits. And in any case, if you're sensible, the baby dino has been born in a secure and sterile environment. This is a creature adapted to live in a world that disappeared 65 million years ago or more. Undoubtedly, there were bugs and diseases around in those times, but they weren't the bugs and diseases of today. Then again, a young creature kept scrupulously away from all possible sources of disease from the moment of birth will, in all likelihood, fail to develop a fully functioning immune system, and will be all the more vulnerable as an adult. Early exposure is to be preferred because it can generate a strong immune reaction. And there's another problem. Our bodies are full of bacteria, viruses, and even parasitic skin mites, most of them either harmless or actually beneficial. Our intestines in particular harbor a whole mini ecology of bugs, bacteria, yeasts, single-celled organisms called protozoa, that has become essential to our digestion. A newborn human baby, like any other newborn creature, has none of that internal complement of microorganisms. But even though parents take some pains to keep their baby safe and clean and away from germs, the parents themselves are repositories of microbes that quickly find their way into the new child. And that's a good thing. Amber-preserved insects, mites and midges might provide some information on what sort of bacteria and other germs inhabited the Jurassic world. But it would be impossible to reconstruct the entire Jurassic ecology down to every last virus, bacterium and dust mite. But perhaps that's not necessary anyway. There's good evidence that lots of insects and microorganisms have been around for a long time in something pretty close to their modern form. So maybe, instead of fussing over your little baby dinosaur and keeping it safe from every conceivable danger, you would be better off raising your new charge in a reasonably clean but not surgically sterile environment and letting nature take its course. Inevitably, that means you're going to lose some number of baby dinosaurs to disease, but in the long run there's really not much you can do about that, especially if you want to run a park where dinosaurs are roaming free. To begin, you have to figure out how to feed your little creature. If it's a vegetarian dinosaur, you can try starting it out on some plain mush of corn or oatmeal, maybe even some mushed up prehistoric plants, if you've managed to cultivate any. But if it's a T-Rex, it's going to want meat from the get-go. Maybe T-Rex's mother would feed it regurgitated bits of prey, the way most birds feed their young today. Or maybe she would just drop a live mouse into the nest and let Junior catch his dinner. It won't be too difficult to know if your dinosaur is a meat-eater or a herbivore. After all, paleontologists have generally been able to figure that out just from fossil remains by studying the construction of the jaw and mouth. Even if it's a species you don't recognize, you can tell from its face and jaw what sort of things it's likely to eat. Then you can just give it a selection of foods and see what it goes for. It's going to be hungry after all, and if newborn babies of all animals instinctively know one thing, it's how to eat. If the vegetarian dinosaurs had eaten leaves and grasses of the Jurassic era, they could probably do just fine on modern leaves and grasses, as long as you exclude anything you know to have toxic substances in it. For a carnivore, maybe you'd throw in some raw hamburger or fresh-killed mice. But would he rush to eat it or stare uncomprehendingly? Perhaps he's expecting to get some chewed-up, partially digested meat direct from its mother's mouth. Well, you've devoted a lot of time and effort to this project, and it's no time to get squeamish. So you chew on some raw meat for a while and spit it out. And you breathe a sigh of relief as the infant begins to eat. Or maybe not. Maybe it wants actual live food. So you throw a live mouse into the dinosaur's cage and see what happens. One of these strategies, you have to hope, will get the dinosaur's attention. First problem solved. It's eating, and you've figured out what to feed it. That doesn't mean your difficulties are at an end, because for all you know there may be some strange mineral additive or vitamin that the dinosaur needs. The only consolation is that deficiency diseases tend to be slow-acting, which gives you some time to figure out what trace chemical is absent from the dinosaur's diets. At any rate, your baby dinosaur is eating, and you can turn your attention to other issues. Disease will probably be the biggest worry and the main cause of infant mortality. To some extent, it's just a matter of common sense in doing your best to keep the dinosaur away from any obvious sources of bad food. You might also feel strongly about keeping them away from any of your lab technicians who've come down with a cold, but that might not be such a big deal. After all, you can't give your dog a cold, and your dog can't give you distemper. It's conceivable, on the other hand, that certain modern germs have the capacity to infect and harm the dinosaurs now revived in your lab. Sooner or later, though, you're going to have to let your new dinosaur have contact with the outside world, with all the dangers, known and unknown, that entails. That's pretty much what the Lost World scientists did in the end, apparently. Having raised dinosaur young as best they could, and got them past the immediate dangers of infancy, there wasn't much alternative except to let them loose on Isla Sorna and allow nature to run its course. Not that there's anything very natural about a lot of dinosaurs scurrying about on a Caribbean island. There's another kind of problem that can afflict the dinosaurs. As Henry Wu remarks, he and his scientists sometimes found that an animal grows for 6 months and then something untoward happens, and we realize there is some error. He means specifically an error in the dinosaur genome that was used to create the animal. You might think that an error in the genome would prevent an egg from growing properly in the first place, and in some cases that'll be right. But there are other errors that could indeed only show up later on, after the animal's born, but before it's fully developed. Whether it's likely that some random problem with the genome would have this sort of subtle effect is another question. It all depends what sort of error there is in the genome. An error, of course, can be small or large. It might be as minor as one single base in the entire genome being incorrect, or it might be as large as a whole chunk of genome being missing or duplicated. What's more surprising is that the seriousness of the consequences that such errors have can be quite unrelated to the magnitude of the error itself. Sometimes a single letter change can be fatal. Other times, a whole chunk of one chromosome can be doubled up, and you might never know. The consequences of errors vary from total insignificance to fatality in an almost completely unpredictable way. This makes some of what Henry Wu says a little hard to believe. He implies on a number of occasions that once they've got the genome figured out, they can tinker with little bits and pieces of the genetic code as if it were just a matter of cleaning up a few bugs in a piece of computer software. At one point, Wu comes to talk to Hammond bearing a file marked version 4.4. This file contains, evidently, Wu's redesigned genomes for a new generation of dinosaurs that would be more to his liking. We could easily breed slower, more domesticated dinosaurs, he tells Hammond. Unless Wu's a whole lot more brilliant than anyone suspects, there's simply no way he could tinker with genomes to make his dinosaurs more domesticated. There is evidence, to be sure, that some aspects of human behavior have genetic roots. There's a mounting belief among scientists that human babies tend to be, for example, either shy or outgoing from the outset, so that their fundamental attitude is inbuilt. But as a baby grows into an adult, his or her behavior changes, depending not just on genetics, but on the way the parents raise their child. It's true that in a general way, some animals tend to be docile, while others are aggressive. Even after thousands of years of genetic engineering the old-fashioned way, by careful breeding through the generations, dogs and cats behave in fundamentally different ways. A gregarious, group-loving animal like a dog can't be turned into a solitary creature such as a cat, no matter how much selective breeding is done. So if raptors really were mean, cold-eyed killers, Henry Wu would not have been able to tweak a couple of genes here and there to turn them into loving pussycats. That bigger change is beyond the bounds of the raptor's fundamental nature. Henry Wu's enthusiasm for genetic tinkering leads him into one last error. To make sure the dinosaurs can't survive away from Jurassic Park, he alters their metabolism in a subtle way. He explains to the skeptical Malcolm that he's made them lysine dependent. Unless they get a rich source of exogenous lysine, they'll go into a coma within 12 hours and expire. Now, lysine is an amino acid, and it's an essential component of certain proteins. What Wu says he's done is to give all his dinosaurs a single faulty gene, so that they can't make lysine for themselves, and have to get lysine supplements in their food, otherwise they'll die. Clever idea, don't you think? Well, maybe not so clever. As a matter of fact, you and I can't make lysine either and have to get it from our diets. What's more, essentially all animals in the world today are in the same fix. None can make lysine. Of course, we don't know for sure how it was with dinosaurs, but the chances are they too got all their lysine from the things they ate. And they could get all the lysine they needed from whatever they ate in the course of a day. Okay, so then instead maybe Wu could have genetically altered the dinosaurs so that they failed to digest lysine correctly. But that's a tall order. There's a whole complicated network of biochemical reactions that extract lysine from food. Some of these steps serve a number of different purposes, so if you disrupt the network, you'd cause more damage than you bargained for. And anyway, if you prevent the dinosaurs from properly absorbing lysine, how are you going to give them lysine in a form they can use? With regular injections? Even Muldoon, the rugged park gamekeeper, would balk at the idea of visiting the tyrannosaurs with a giant needle every day. When Wu says that the way they figure out what a particular genome represents is to grow it and find out what it is, he's saying more than he realizes. All his other remarks about fine-tuning the dinosaurs to behave in a certain way or be dependent on lysine supplements are implausible, but even more problematic is that for all his precautions and research, Wu's only way to find out if and how his dinosaurs would survive in the wild is simply to throw them out of the lab and hope for the best. As with raising children, there's only so much a parent can do. Sooner or later, the offspring are on their own and have to fend for themselves. Eventually, there will come the time when you proudly let the dinosaurs you've raised loose to romp through the spacious parklands you've prepared for them. Pretty soon you'll have an authentic-looking Jurassic world inhabited by dinosaurs. Well, maybe. Another possibility is that you let your dinosaurs loose and they all starve or die of exposure. Then again, most animals have a pretty strong urge to survive. Instead, they eat up a storm. In short order, the plant-eaters denude the park of vegetation, the meat-eaters gobble up the plant-eaters at will, and then everyone dies, having laid waste to the lavish park you set out for them. Your young dinosaurs represent billions of dollars and years of effort. You're not going to just let them loose in a field and hope they'll figure out how to survive. There's no one except you to teach them how to eat, where to hide, what to chase, what to run away from. Your dilemma is that at the same time you'd like to avoid human contact as much as you can because you want the animals to grow up as dinosaurs and not as pets, or very confused creatures who aren't at all sure what they are. The nursery for the dinosaurs of Jurassic Park turns out to be the secret island, Isla Sorna, Site B of the Lost World. There, because they can't figure out how to raise the dinosaurs they've created, Henry Wu and his team more or less give up the effort and let the animals loose. A sort of hastily enforced evolution takes place. If the animals manage to survive, they can be transported to Isla Nublar. If not, too bad. You can always make a few more dinos and try again. The dinosaurs of Site B display a variety of dysfunctional habits. The nastiness of the velociraptors is put down to a lack of parental guidance. Raised without any constraints, they become reckless carnivores, chasing prey at every opportunity and not always eating what they catch. The raptor's home area is made to sound like something out of the movie Animal House. What's more, the raptors are mean to their own offspring. When a pack of raptors has brought down a large animal, the whole herd descends on the corpse in a feeding frenzy, and young raptors have to fight their parents for scraps. Unlike the raptors, the triceratops of Site B turn out to be admirable parents. When the herd is under threat, the adults form a ring around the youngsters, protecting them. The apatosaurs have even worked out a bit of cooperative behavior with the parasaurs. The bigger apatosaurs can defend themselves with swipes of their heavy tails, but they have poor eyesight. The parasaurs, on the other hand, see danger quickly but are smaller and more vulnerable. So the two herds intermingle, the parasaurs providing the warning signals and the apatosaurs acting as the defense. But if the raptors have so dismally failed to learn any manners, how would the triceratops, apatosaurs and parasaurs do any better? Behavioral characteristics may well have genetic roots, but even so, whether or not those characteristics develop depends a great deal on the actions of parents and on the general environment. Behavior in animals as well as humans emerges as an inseparable mix of innate characteristics and learned behaviors. The orphan dinosaurs raised entirely in the lab may well display some sort of social behaviors when they are released into the wild, but there's no telling how those behaviors would have been modified, guided, and changed in the social structure of a true dinosaur community back in Jurassic times. In Jurassic Park, incidentally, all the animals are supposed to be females to prevent the herds from reproducing. In the Lost World, on the other hand, there are obviously males and females, since dinosaurs on Isla Sorna have been living and breeding for 5 years before Malcolm and the others rediscover it. It's not clear whether some of the Lost World dinosaurs became male, as they did on Isla Nublar, for the dubious reason that bits of frog DNA in their genomes made them transsexual, or whether Henry Wu and his team were making males and females all along. In any case, a purely female, or purely male for that matter, herd of dinosaurs would be a strange and unpredictable thing, since in most social animals the battle of the sexes enters into a lot of the cooperative or aggressive behavior. The raptors of Jurassic Park and the Lost World are an odd case. They're greedy, selfish and aggressive, even toward each other and their own infants. On the other hand, they're also said to be skillful pack hunters, which means they must put their rivalries aside while chasing their prey. But how did the orphan raptors acquire the cooperative skills needed for hunting, while remaining completely devoid of the similarly cooperative skills needed for living and surviving in a pack? In particular, the idea that raptors would deny food to their own infants is difficult to believe. Most animals display an instinctive protectiveness toward their offspring, their own offspring at least. Who knows what basic survival skills mother dinosaurs or a herd of adults would have taught their youngsters? Surprisingly, paleontologists have been able to make a few simple inferences about the way some dinosaurs raised their young. Jack Horner, digging up the bones of great herds of myosaurs on the North American plains, discovered nest sites, and discovered too that the bones of juveniles up to a moderate size remained at or near the nests. It seems reasonable to guess that the parents kept their offspring near the nest and fed them until they had grown to a certain size. Paleontologist Mark Norell and his colleagues have found fossilized raptors of various kinds apparently sitting on nests to safeguard their eggs, meaning that even the predatory raptors were good parents. The plain fact, though, is that even if some sort of stable dinosaur society does emerge, you won't really know whether your mini ecosystem reflects the way real dinosaurs would have lived tens of millions of years ago. Moreover, you're putting together a variety of dinosaurs from different eras, and you're forcing them to live in a confined space. In reality, some of those creatures that were contemporaries may have learned to stay well out of one another's way. In Jurassic Park and on Site B, they don't have that option. Since you have to put the dinosaurs in a small space, the first thing you want to be sure about is that it's enough space. Isla Nublar measures 8 miles long by 3 miles at its widest point. According to Hammond, the total area of Isla Nublar is 22 square miles, but there's obviously a certain amount of space taken over by the visitor center, the research labs, and so on. Let's be generous and suppose that something like 20 square miles is open for the dinosaurs to roam in. That's equivalent to an enclosure about 4.5 miles on a side. Sounds pretty big, you might think, but then some of the dinosaurs are pretty big too. In Montana, Alan Grant and Ellie Sattler are digging up dinosaur fossils in an area where they found almost nothing except hadrosaurs, which were large grazing animals. They estimate from the numbers of remains that the hadrosaurs roamed over ancient North American grasslands in herds of 10 or 20,000. The reason Grant and Sattler find endless numbers of hadrosaurs but little else, they decide, is because the predators must have been much rarer. A herd of 10,000 hadrosaurs would have supported a population of only about 25 tyrannosaurs. More than that, and the hungry meat-eaters would thin out the hadrosaur population faster than they could reproduce. There's not nearly enough room on the island for 10,000 hadrosaurs. But 100 hadrosaurs, going by the numbers Grant and Sattler guessed, would support only 1/4 of a T-Rex, which, needless to say, wouldn't exactly form a self-sustaining population. Looking at things the other way around, you might decide that you have to have at least a couple of dozen tyrannosaurs to form a viable population. Any less than that, and they would be susceptible to disease, wiping them all out. But to sustain that many tyrannosaurs, you'd need tens of thousands of hadrosaurs, and for that you'd need a Jurassic Park the size of Connecticut. Hammond and his team get around this problem on Jurassic Park by making the island into what is basically a zoo. The animals are confined to different areas, and they are fed. This is not a functioning ecosystem, even in an artificial way. The tally of animals on the island includes 2 tyrannosaurs, 8 raptors, and 7 dilophosaurs. That's 17 hungry meat-eaters in a population of just 238 animals altogether. Left to their own devices, the carnivores would probably polish off all the other animals in a matter of weeks or months, then fight it out with each other. But if you're feeding the animals rather than letting them get their own meals, you've got a lot of feeding to do. In one scene, a hapless goat is pegged to a stake somewhere in the T-Rex area, and the ground begins to shake under T-Rex's approaching footsteps. Pretty soon, no goat. How many goats a day would constitute a healthy diet for a full-grown T-Rex? Here's a chance to do what scientists like to call a back-of-the-envelope calculation. A person weighing 200 pounds will eat a few pounds of food every day. So let's say a typical animal needs to consume about 2% of its own body weight every day, 4 pounds of food for our 200-pound person. T-Rex is thought to have weighed around 15,000 pounds. 2% of that is 300 pounds. How much does a goat weigh? Let's say 75 pounds. It looks as if we need around 4 goats every day to keep Mr. T-Rex happy. Okay, 2 tyrannosaurs, 8 goats a day. The raptors are smaller. Let's guess they weighed about 500 pounds each, in which case 1 or 2 goats every day would keep the pack well fed. Then there are the dilophosaurs. Let's throw in another couple of goats for them. Altogether, you're looking at around a dozen goats every day. If you had a farm on the island, you'd need a pretty substantial size herd to be able to take a dozen every day, maybe as many as 1,000 goats. Since you're squeezed for space, you'd have to ship the goats in, which is what Hammond implies they do, except he also explains that the sea passage can be dicey because of the weather. You couldn't rely on daily goat deliveries, so you'd have to have some sort of pen in the island where you could keep a few dozen goats in case the ship didn't get through. Maybe things wouldn't be so difficult for the plant-eaters, though. At least they can graze in the meadows of Isla Nublar. Unfortunately, Hammond has put 17 apatosaurs in the park. Now these guys weigh 20 to 30 tons apiece, which is equivalent to almost 100 good-sized dairy cows. So just in terms of feeding the apatosaurs, you have the equivalent of something like 1,700 cows on your island. You can keep a small herd of 20 cows on a few acres of grass. A square mile is 640 acres, so the herd of 17 apatosaurs looks as if it will need a square mile all to itself, at the minimum. Then there are 21 myosaurs, which represents another 300 or 400 cow equivalents. Another good piece of a square mile for them, therefore. And there are 11 hadrosaurs, 33 hypsilophodontids, 8 triceratops, and assorted smaller animals. It's starting to look as if the whole island is getting taken up with meadows and grasslands for the plant-eaters, and we've still got to find room for the carnivores and for the goat farm to supply them. And let's not forget that the island is described as a rugged and craggy place with forested slopes. So maybe you have to have grass and hay brought into the island too, along with the goats. This is getting difficult. You seem to need a pretty big ferry coming over every couple of days. Site B, the Lost World, is a different proposition. There, things have been left to go wild for at least 5 years by the time the good guys rediscover the island, and when they arrive, they find a lush place teeming with life. What's more, the island itself seems to be in pretty good shape, with plenty of thick vegetation, trees, and meadows. There are as many, if not more, animals on Isla Sorna as there were on Isla Nublar. The dimensions of Site B are not directly given, but judging by the map and the distance Levine and the others travel in their jeeps, it's somewhat bigger than Isla Nublar, perhaps 100 or more square miles. Even so, it's arguable whether Isla Sorna is really big enough for the animals on it to last 5 years without killing each other off in large numbers or destroying the landscape and vegetation. There's also the problem that there are too many raptors, not to mention a number of tyrannosaurs, for the few hundred other dinosaurs on Isla Sorna to support. This is a point that puzzles Malcolm and Harding. 12 raptors, which is what they find, would have made much bigger inroads into the herbivore population than they seem to have done. The explanation can't be that there were lots more herbivores originally, because the herbivore population is already as much as the island can bear. What Harding and Malcolm decide is that the animals suffer from an epidemic disease that makes them die young. This disease is none other than a version of bovine spongiform encephalopathy, better known as mad cow disease. The dinosaurs were supposedly fed ground-up sheep meat, which contained protein fragments called prions, thought to be responsible for BSE and other related diseases. Infected meat gives the dinosaurs a BSE-like disease. The raptors die young, and, Harding explains, the rapid die-off supports a much larger predator population than you'd expect. Unfortunately, this explanation makes no sense at all. It may be that the raptors die young, but when the visitors are on the island, there are nevertheless 12 raptors. 12 raptors will eat what 12 raptors will eat, no matter what their life expectancy happens to be. What this all comes down to is that Isla Sorna is just too small to support the thriving population of dinosaurs that are supposed to have lived there for 5 years. If you really wanted a place where you could set dinosaurs free and have them live and breed at will, you need a bigger island. Where are you going to find one? Apart from space, you also need to worry about climate. It's not just a matter of finding a climate that the dinosaurs will like, because the dinosaurs on Jurassic Park didn't all live in the same conditions. In fact, they didn't all live in the same geological era. If Hammond had really populated his park the way he describes, he would have been mixing up dinosaurs that didn't live within 100 million years of each other. That makes it tough to find one place, especially a single island, that can accommodate all the different creatures. Islands are hard to come by these days. Anything substantial has been explored, mapped, colonized, or otherwise taken over by one country or another. It's hard to say just how big your island will have to be, but if you think of something at least 10 times as big as Isla Nublar, covering at least a couple of hundred square miles, you're getting into some large chunks of real estate. Barbados, for example, checks in at 166 square miles, and the French island of Martinique at just over 400 square miles. Buying an unknown, uninhabited island off the coast of Costa Rica is one thing, but trying to take over an entire country is another matter altogether. The chances of even John Hammond being able to lay his hands on an island that's big enough and has a suitable climate seems unlikely, to say the least. That leaves a mainland park, with all the additional risks that would entail. There's another fact we've overlooked so far. Any piece of land that's suitable for dinosaurs to live will already have a full complement of modern wildlife. Not just plants, insects and bacteria, but birds, lizards, rats, monkeys, rabbits, who knows what. Do you scour every large animal from the park before you install the dinosaurs and hope that the dinosaurs themselves can establish their own replacement ecosystem? That's unlikely to work. Dinosaurs in their original environment coexisted with a huge variety of other creatures, and may even have depended on them in some ways. If you wanted to recreate a true Jurassic landscape, you'd have to find DNA for every single creature that lived and make copies of those animals too. That's not going to be possible as a practical matter. So the only thing you can do is allow the wildlife that's already in your dinosaur preserve to stay there, and hope it can establish some sort of balance with the dinosaurs. This is evidently what must have happened at Site B, although little mention is made of any non-dinosaur creatures that inhabited the island. But what results is even less authentic a dinosaur system than we had already bargained on, since it ends up being a composite of dinosaurs from different climates and eras, along with whatever modern plants and animals happen to be around. Ian Malcolm's belief that by studying Site B he can learn clues to dinosaurs' true behavior is not at all believable. We simply don't know, in any precise sense, how dinosaurs moved, what sort of mating displays they might have made, whether they curled up to sleep like cats or hunkered down like cows, and so on. This gives Michael Crichton a good deal of license to invent seemingly realistic scenes of animal life on the two islands. One of the great attractions of Jurassic Park and Site B would be the range of sights, smells and sounds that fill both places. Much of the atmosphere of the books and movies comes about because the fossilized skeletons of dinosaurs have been given flesh and colors and noises. These dinosaurs are green and brown and red and yellow. They honk and roar and trumpet. They have bad breath, runny noses, and poisonous spit. On the one hand, most of this is entirely reasonable. The dinosaurs were highly evolved animals, and no doubt had colors, noises, and behaviors to rival what we see in the modern world. On the other hand, if we want scientific evidence for the way any particular dinosaur looked, smelled, or sounded, we draw almost a complete blank. In a very few cases, impressions of skin have been fossilized, and we can see that these dinosaurs had some sort of leathery, thick, dimpled skin. But as to what colors the dinosaur skin might have been, what precise noises they would have made, for that, you might as well just make it up. You can have some confidence that big animals make louder and deeper noises than little ones, but that's about it. And as for color, we know exactly nothing. Traditionally, they've been portrayed in browns and greens because they were thought of as large reptiles. But if you think of them as precursors to birds, a whole new range of colors comes to mind. Why not give the dinosaurs some bright colors and arresting noises? Until somebody clones a real, live dinosaur, nobody can prove you wrong. Dressed in black and constantly warning of disaster, the mathematician Ian Malcolm is perhaps meant to represent the conscience of Jurassic Park, the person who's always asking whether John Hammond and his genetic engineers are doing the right thing. As always happens in books and movies of this sort, nobody pays much attention to him until disaster actually strikes. The movie is pretty much your standard thriller, in which the good guys eventually triumph and the bad guys and the dinosaurs are defeated. In the book, on the other hand, Michael Crichton conveys a much stronger message about the tendency of scientists to do things just for the sake of doing them, and of the parallel tendency of things to go wrong because, in practice, everything turns out to be more complicated, more difficult, and more unpredictable than the scientists had bargained for. Ian Malcolm goes so far as to argue, constantly and irritatingly, that he knows for a fact that disaster is bound to strike. It's not just that things go wrong, according to Malcolm, but that chaos theory, the mathematical theory of complex systems to give it a more formal title, actually predicts that things will go wrong. Chaos theory is supposed to be about the inherent unpredictability and randomness that ensue when, say, you throw a bunch of dinosaurs together in a time and a place that are alien to them. Malcolm can't predict exactly what will go wrong, but according to him, chaos theory proves that something will go wrong. Malcolm dresses up his dire warnings with a lot of fancy mathematics and obscure jargon, but the basic idea is one we all know by one name or another. It's the well-known principle called Murphy's Law. If something can go wrong, it will. Surely, you might think, there must be more to chaos theory than a fancy-dress version of your grandparents' folk wisdom. Well, there is, of course, but it's not clear from the book just how much Malcolm really knows about chaos theory, or about elementary mathematics, for that matter. For example, when the visitors to Jurassic Park are first being shown some of the computer systems that keep track of dinosaurs on the island, they get to see a graph of dinosaur infant mortality through about 1.5 years of the park's existence. The numbers spike up and down from month to month, and Malcolm remarks this alternation is characteristic of many complex systems. The idea he's hinting at here is that in systems where a great many variable factors influence the outcome, whether it's dinosaur mortality or water dripping from a tap, complexity results in unpredictability. Instead of a nice steady drip, 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 as Malcolm puts it, you get an irregular sequence of little drips and big drips, with seemingly random pauses in between. In the same way, Malcolm wants us to believe, the irregular pattern of dinosaur infant mortality is the result of a complex, chaotic system at work. But if Malcolm tried to make this kind of argument in a scientific seminar, he'd be laughed out of the room. The main problem here is that the numbers are too small to allow any big deductions to be made from them. This sort of variation can easily come about by pure statistical chance. Even in a perfectly orderly system, the number of infant deaths is bound to vary from month to month. And especially if the numbers are small to begin with, that variation can be quite large. So there's really not much cause for Malcolm to make any sweeping generalizations about chaos and complexity from the one little graph he's presented with. There's an even more elementary mathematical error that Malcolm makes. The computer that tallies the animals in the park can also check up on their sizes, and it presents to Malcolm and the others a graph showing the distribution of heights among the compies. Some are smaller than average, some larger, but most of them cluster around the middle of the height distribution, giving the standard bell curve. Malcolm first makes a correct observation. If the lab-created compies were born in 3 separate batches at 6-month intervals, as Arnold explains, then you wouldn't expect a bell curve for the population as a whole. Malcolm indicates the sort of 3-hump distribution of heights you'd expect, because there are in effect 3 distinct population of compies of different ages, each one characterized by its own bell curve. That part of Malcolm's argument is fine, so far as it goes, but he misses a much more elementary point. The total number of compies on the island is, apparently, 65. If you were to measure the heights of 65 adult people, randomly selected, you would assuredly not find a smooth, perfect bell curve of the type that the computer depicts. If you measured the heights of 650 people rather than 65, you'd expect something much closer to the mathematically ideal bell curve. Measure 6,500 people, and it'd be closer still. But measure the heights of just 65 compies, and you'd get a rough, approximate, wiggly version of the bell curve, not the perfect thing Malcolm is shown. The fact that he doesn't pick up on this point hints at a weak spot in his mathematical knowledge. Things get even more confusing when Gennaro, the lawyer from the InGen Corporation, asks Malcolm to explain why he thinks dinosaurs must have escaped from the park. The mathematics are so self-evident they don't need to be calculated, is his unhelpful answer. It's a pity he doesn't give some idea of what his calculations were, because the idea that he can prove mathematically that animals are bound to escape from a nature preserve such as Jurassic Park would be a notable discovery. What Malcolm seems to be arguing is that the designers of Jurassic Park are attempting to recreate an entire functional ecosystem, and that because ecosystems are inherently complex and unpredictable, it's inevitable that things will go wrong. Part of that argument is reasonable, but leaping from there to the idea that dinosaurs are bound to escape takes some doing. In one sense, Malcolm is quite right. the designers of Jurassic Park would be foolish to imagine they could recreate an authentic dinosaur ecosystem and expect it to remain calm and stable. But Hammond and Wu acknowledge this problem openly. In Jurassic Park, some of the animals have to be kept separate from others, and food for all of them must be brought in from the outside. So there's no question that Jurassic Park is more like a large zoo or a wildlife park than a true recreation of dinosaurs in their natural habitat. And if you're keeping track of every animal, feeding them, separating some in large enclosures, and so on, you have as much control over the animals as your resources and your competence allow you. What Malcolm's musings have to do with dinosaurs escaping is another question altogether. Chaos theory might imply, in the case of Jurassic Park, that Hammond and his crew would find it impossible to maintain their dinosaur populations at constant, equitable levels, for the simple reason that constant, equitable levels don't exist. But even though changes of this sort may be unavoidable, that doesn't mean any old thing can happen. You might well have chaotic behavior in terms of the numbers of animals, or their rate of birth, or the frequency with which they eat other dinosaurs or are eaten by them. But this kind of chaos remains bounded by external constraints, most notably the fact that the dinosaurs are on an island. Chaos theory couldn't predict that the raptors would escape by hiding in a boat. In principle, if you put enough barriers around the island, you can keep the dinosaurs in. It's true that dinosaurs do manage to escape, but the reason has nothing to do with chaos theory or higher mathematics. It's because the security fence has gaps in it, and because Dennis Nedry, the saboteur, turns the power off. There's no way Malcolm could use his theories to predict the presence of a saboteur, and Nedry's actions clearly go beyond the bounds of ecosystem theory. On the other hand, there's an easy way to predict Nedry's role in the story. There has to be a bad guy. That's completely predictable, and therefore has nothing to do with chaos theory. In The Lost World, Ian Malcolm has a bigger theme. Chaos theory, also known as the theory of complex systems, can explain how dinosaurs became extinct. Along with that are asides about the significance of complexity as an extra ingredient in evolution that must be taken into account along with traditional elements of biology. As usual, it's a bit hard to decipher Malcolm's pronouncements, but we can sort out two main themes. The first has to do with the difficulty that many people have in understanding how evolution can lead to such superbly specialized biological structures as eyes. The argument is a modern variation on the old argument from design, by which philosophers and theologians wanted to prove the necessity for a divine being. The eye is so complex it's impossible to understand how evolution could have produced it, therefore someone must have designed it. Malcolm gives a version of this argument, but hastens to deny he's any sort of creationist. Instead, he hints mysteriously, other forces are also at work. The other forces, needless to say, come from complexity theory. In particular, he talks about the idea of self-organization, which is what's supposed to happen when complicated interacting systems, made of many interconnected parts all doing their own things, manage to fall into relatively stable forms of behavior or activity. This idea is sufficiently vague and inarticulately stated that it's possible to think there may be something to it. The main difficulty, however, is that Malcolm is struggling to resolve a problem that most biologists agree doesn't exist in the first place. Even a primitive, poorly functioning eye, even a single cell that can tell light from dark, would be of use to an organism that previously had no sense of vision at all. And if a single light-sensing cell is useful, then a group of cells that can distinguish degrees of light and dark would be even more useful. It is possible, in other words, to imagine a series of progressively more complicated and efficient light-sensing structures being built up, one tiny improvement at a time. And as for this business of self-organization, it should be emphasized that evolution is precisely the result of genes working together. One gene influences another's action. Many genes working together form units that have some common purpose or function. If you want to call this sort of thing self-organized behavior, then evolution already has it. Malcolm's theory about dinosaur extinction, as far as we can figure it out, is that dinosaurs were doing just fine, but that at some point the complexity of their interacting ecosystem began to sputter forth unpredictable disturbances in the behavior of the animals, which ultimately led to their demise. Malcolm's point is that changes to the ecosystem need not be beneficial. It's possible that after a sufficiently long time, some abrupt and unpredictable thing could have happened that pushed them into extinction. Quite what that change might have been is hard to guess. The point about changes in complex systems is that you can't pinpoint a specific cause in the conventional sense. It's the same old system that it always was, and just happens to have stumbled into an unstable pattern of behavior. Despite everything Malcolm says, it's hard to see how some change of behavior could have been so large and abrupt that the dinosaurs went from prosperity to extinction in barely 1 million years at most. And it's equally hard to see how Malcolm hopes to learn anything useful about dinosaur behavior by studying relatively small numbers of animals unnaturally introduced into a crowded and artificial environment. In any case, Malcolm's theory is sharply challenged right at the beginning of The Lost World, when Berkeley paleontologist Richard Levine is listening to Malcolm give a lecture on his work. Levine observes that dinosaurs apparently became extinct all around the world at the same time, and asks why, if unpredictable changes in dinosaur behavior led to their demise, those changes should have happened everywhere, in many different places and environments, all at once. A good question, and one to which Malcolm never provides an answer. In 1944, scientists figured out that a molecule called DNA carried hereditary information. In 1953, James Watson and Francis Crick worked out the structure of the DNA molecule and thereby explained the means by which it replicates. Today, scientists have the ability to manipulate little pieces of DNA, genes, and thus affect for good or ill the genetic constitution of whole animals. Much experimentation has been done on plants, bacteria, worms, and fruit flies. Mice have been deliberately created with genetic disorders that mimic similar problems in human beings, in the hopes of finding a solution to those human disorders. Plant breeders have been manipulating crops for centuries by crossing different varieties to create new forms, more resistant to disease, more tolerant of drought, more productive. But now scientists can put genes from one plant species into another, the equivalent, if it were possible, of interbreeding tomatoes and bacteria. To scientists, this kind of precise genetic engineering may represent the inevitable refinement of processes that humans have been engaging in ever since they became farmers. But to the general public, the idea of adding and cutting out single genes to control highly specific aspects of a plant or an animal, and even more, the insertion of genes from one species into another, sometimes seems the first step in transforming a frightening science fiction story into reality. One day, scientists hope, it will be possible to correct such common and disabling genetic disorders as cystic fibrosis or sickle cell anemia by deleting or inactivating the faulty gene in a person's genome and replacing it with the correct version. Who could argue against such a cure? In the meantime, we find ourselves frequently in the position of being able to detect such disorders without being able to cure them. Predispositions to various cancers and other life-threatening diseases can now be detected in a fetus. Parents can be screened and told that their children are at high risk for this or that problem. That may leave parents with some difficult choices, but at least the scientific facts are unambiguous. But what happens when all a doctor can tell you is that your child will have an increased risk of some heritable disease, but may never develop it? What if a child will be a few inches shorter than average, or has a tendency to obesity? Many of the factors that genes influence may be conditions that society deplores, but that are also a normal part of the variable makeup of the human race, not medical disorders in need of correction. The ability to understand and interpret the genetic code has the potential for good and bad, something often true of developments in science. And when it comes to making ethical or moral decisions, scientists are no better or worse than the rest of us. As these new forms of knowledge become public, the public will have to ponder some weighty matters that may make the question of whether or not we ought to recreate dinosaurs seem trivial. You'll have realized by now that making dinosaurs from ancient scraps of preserved DNA is no easy matter. It may simply be that no one will ever find any dinosaur DNA at all, or never any more than a few scraps of it. And even if sufficient dinosaur DNA is found, there are countless difficulties and complications in creating a dinosaur, enough to make the task essentially impossible. But the sorts of problems that remain perplexing, how to identify and mend unknown genes, how to persuade an egg cell to take up a foreign genome, how to manipulate the genetic characteristics of a growing embryo, are those that scientists are working on right now in many different contexts in laboratories around the world. The work that's been done up to now in creating genetically engineered organisms and releasing them into the environment is modest compared to making a dinosaur, but nevertheless it's been a battleground both legally and politically. Some years ago, researchers in California inserted a cold resistance gene into Pseudomonas bacteria. Eventually, these engineered bacteria were sprayed onto strawberry plants in open fields in order to ward off frost damage. But the enterprise had to overcome enormous opposition, particularly because this was the first deliberate attempt to release altered genetic material into the environment. But in matters of genetic engineering, it's public opinion that seems to win the battle in the end. If frost-fighting bacteria make strawberries abundant year-round, it's likely that concern about possible technological ill effects will fade. If it ever does seem possible to revive dinosaurs, public enthusiasm may make it impossible for cooler regulatory heads to hold the project up, or even debate its ethical implications. Undoubtedly, there will be some people who deplore the notion of a Jurassic Park, who find it offensive, for one reason or another, for scientists to rebuild what nature long ago rendered extinct. Others will decide the project is too dangerous to attempt. But John Hammond was surely correct in thinking that huge numbers of people will find the idea, whatever perils it may bring, so fascinating, so awe-inspiring, that they will provide enthusiastic and essential support. 50 years from now, it may be that many of the technical obstacles to dinosaur recreation, obstacles that seem insurmountable today, will have been overcome through the efforts of scientists pursuing all kinds of other goals. Yet, if we ever reach the point where we have the technical ability to create dinosaurs, we may find that the ethical problems involved in resurrecting an extinct creature will pale in comparison to the quandaries we will encounter because of our ability to manipulate the tiniest genetic elements of living creatures, most particularly ourselves.